I want to highlight some of the characteristics of research and how some of the work you've already done with the CHIP process, kind of how we could almost transition that into a framework of research through just a few little changes. Talk about the phases of research transition, provide a little framework for the levels of community engagement so that as you guys start engaging with academics, you kind of have an idea of where you'd like to be in that partnership and how you'd like to share information and collaborate with the academic partners. And then just give an overview of our CHIP to CHIRP training, so really to highlight how we've structured some of these curricular elements to really map onto the research project. So what we heard, I know some of you just participated in the intake surveys this morning, but we really heard two things, and that's that people were interested in skill development. They wanted to know really, how do you do research on humans? They might have heard about how you do research on animals, but how do we collect information in our population level? Or some of you who work, for example, in public health or other agencies, you might have programs, and so people had questions. One person said, you know, I do outreach in the Latino community and I don't know what goes on behind the scenes to develop programs through research. I'd like to learn more. So people thought this was a real opportunity to build skills. We also heard strongly that people want to do things that can impact and benefit the community, and they really see research as one of the ways that your efforts can have a strong impact on the community and that you can evaluate what that impact is. So research is really valuable. This is a cartoon that this says, then a miracle occurs. It's talking about the process and developing the equation, and the professor saying, I think you should be more explicit here in step 2. So part of what often happens is we might have programs that we do in our communities, but we might not actually know what the active element is that makes a program effective, or we might not collect data that allows us to say, you know, this is the real active reason why, say, we were able to improve physical activity, or we were able to increase awareness or performance of certain health behaviors. And so research really gives us a framework and a process to start asking some questions about things we often engage in our daily lives, but in a systematic approach, so that we can really say, it's not just a miracle, but this is one of the reasons why. The thing I just wanted to point out is that we can pretty much address almost any question through research. You could look at how do you punish your child, or what's valuable. You could look at sleep disorders and academic performance. We can look at chronic pain and depression, you know, which comes first, or are they co-occurring, you know, do they actually influence each other? We can look at dog ownership, health and physical activity, you know, so there are all kinds of questions you can look at through research. We can look at childhood obesity and how do we make policies that actually help us reduce this epidemic before it becomes prevalent in people's lives. So is the CHIP assessment research? So how many of you have helped out with the baseline CHIP assessment, where you guys do interviews in the community and you look at data on your kind of current health context here? Great. So what Beth has described is really what we call in the academic community a mixed methods approach, which is where you use qualitative data, which are the stories or the interviews that you guys actually participated in this morning, and you also use numbers or quantitative data, and you really look at the complementary nature and what each of those can tell you about the local context. So the CHIP assessment is really a process, you know, and it's a process to understand what's going on in your local community and what you want to tackle. And it's very similar to what we do in research, but research adds a little bit more structure and rigor and a very clear kind of protocol and set of processes that you're going to adhere to up front, as well as how you're going to analyze and report that data out. Okay? And so we'll talk a little bit more about how we can transition kind of this community health assessments that we do frequently, how you can add the research lens to kind of just make the scientific rigor behind those approaches a little bit more concrete. So what is research? So Webster, you know, he says the dictionary, investigation or experimentation aimed at the discovery and interpretation of facts, revisions of accepted theories or laws in light of new facts, or practical application of such new or revised laws or theories. Or the Federal Regulations Code that really specifies how we go about research and what steps need to be in place and what processes and what the rules are around research. It's a systematic investigation. We're testing things and we're trying to develop generalizable knowledge. So we're trying to collect information here at the local level or at the local context in a way that allows us to hopefully scale up some of what we're doing to other areas, or so that they can use the approach we use to actually address similar issues in their community. So characteristics of research. The CHIP assessment is, kind of a foundational start to how we might do research. But research really, you look at making it systematic, formalized, and really having an explicit process. So you have generally a protocol that you're going to go through, and you have an institutional review board that actually approves that protocol and makes sure that the benefits of participating in conducting this research outweigh the risks. And so they do a concrete review, and we actually will talk more about that and what they're looking for and why, because there's actually been a history of kind of exploitation of some underserved populations through research. Okay, so we want to make sure that doesn't happen. There's a process where you state assumptions, you develop the plan to study, you collect data, you analyze it, and you disseminate it. So you share it either in academic publications or in newspapers or in ways that you think are important to raise awareness about what you discovered. Research has clearly defined concepts, it's systematic. You use instruments and measurements that ideally have been developed through a process, okay? So you don't always just create a questionnaire, you don't just write your questions down and then administer them to people. Ideally what you do is you find questions that have already been developed and validated and shown to be effective in other situations, and you try and use those in your own setting, or you try and modify them slightly through a process for use in your own setting. And then reporting results. So we all see the world, right, from our own lens. And one of the keys that research really tries to do is to give us a framework so that some of our personal biases, they aren't necessarily pushed out onto others, that we use a process that allows us to really see what the data and what the true circumstances are. So I think Paul has maybe talked a little bit about the phases of research, but there's something called kind of stages. And so you think about bench research, and so this is research generally on rats or in the lab. You think of kind of their scientists at their bench, maybe testing a new drug on rats, or you might think of looking at how cancer cells divide, okay? And then you think about moving from bench to bedside. They call this translation 1 or T1. So bedside is actually taking information, so taking medications and trying to test them on humans to see what the effectiveness is, okay? And then once we know that something, so let's say we have a new diabetes medication and we know it's more effective than previous medications were. The next step is, well, how do we actually get that out into practice? And so T2 is really the developing of guidelines or systematic reviews, so looking at the literature and really saying, this is the best approach for treating a patient. When we get into T3 and T4, that's really, how do we get this information about best practices out into the community? So let's say that there's a certain approach for teaching or education that's been really demonstrated to be effective in these controlled environments. How do we actually get them out into Madras or Prineville schools? How do we actually make this practical in the real world? And they really call that translation and dissemination research. And the thing that's really important about translation and dissemination research is that, the context in a rural area is very different than the context in an urban area. And what you really need to think about is, how can we maintain what they've discovered to be effective about these interventions or about these treatments? But how do we also tailor them so that they're appropriate in our current environment and our current context, given who the players are and what things look like? So I just wanted to introduce a few key terms. And so when we think about research, we often start with a research question. So you might start with something like, well, how do school-based gardens influence, child awareness of nutrition and health behaviors? And then what you would do is you would actually refine that research question and state it as a study aim. So you would say, we want to evaluate the impact of participation in a 6-month school garden program on child nutrition habits and interest in consuming new foods as compared to similar peers who did not participate. Okay, so you take your research question, we want to know about this, and we state it in the terms of, we're going to investigate, or we're going to aim to accomplish this goal in our study. And what you might do is you might actually state a few hypotheses. So you might state something like, you think children who participate in the school-based program might be more likely to choose nutritious foods, or they might be more likely to actually be willing to try new foods. And so you really use this process of thinking about the research question, looking at the literature to actually see what's out there, what's come before, to figure out what's the new question you need to ask. Okay? So one of the key things about research is you never have to start from scratch, right? So what you can do is you can see what's come before, and you can see, well, how can the question we ask really build on the knowledge that's already out there? How can we push the field forward through our question? The other thing is study team members, and this was in your pre-assessment, I think. So one of the things is there's a principal investigator on each project, and this is really the person who is generally, they write the grant, they have ultimate authority for how the process unfolds, for the quality of the data that's collected. They're responsible for making sure that we have proper institutional review board approval, and that the approach that's being used is appropriate, and that the protocol is being upheld. You also have co-investigators, so these are other team members that help out, a project manager who's kind of the overall coordinator generally for a project, and then you can have research assistants and a statistician. So in most studies, you generally want to try and get input from a statistician to make sure that you're getting information from the right number of folks, and that you're looking at the right factors that might influence the question that you're asking, that you're controlling for those factors. And I think the key question is, where do community partners fit in? These are kind of some traditional terms, but how do we work with community partners in this process? And the answer is that, community partners can be involved at all levels, and really the question is, how do we help facilitate this type of collaboration? I didn't put statistician because I still go and get help from a statistician. So if you guys are experts, I'm going to start coming to you. So, the other thing I want to talk about is as you're starting to engage in this process of collaboration, one of the things that you might want to think about is, there are different levels at which communities can be engaged in research. Okay, so in some cases you might have people who come out and they might administer surveys in your community, they might collect data. But you might not actually have had a role in determining what the questions were or how that data was collected. You can have kind of lower medium levels of activity, which is like getting a focus group together at the start of a study so we can figure out, well, what's the question that needs to be addressed? What's the important framework that needs to be asked? You can have communities help actually implement a study that was designed by a researcher. So let's say we want to test a new medication. How do we actually get people who are real people in the real world to utilize this? Or you can have a really high level of participation and collaboration in research, which is kind of the framework behind community-based participatory research. And that's the idea that you're really, as a community member, an equal collaborator with the academic partners across all phases of the research process. And I think there are actually benefits and maybe challenges to each level of this kind of phase of research collaboration. But I think the question is just, as we move forward, how would you like to participate in research? So in your perspective, just to think about how you'd like to be a collaborator, how you'd like to partner, and to recognize that there are differences. And one of the reasons there are differences is that some populations have been exploited by research, and so there's this quote, outside researchers swooped down from the sky, swarmed all over, asked nosy questions, and then disappeared, never to be heard of again. Okay, so really the chip to chirp process is about building an equitable collaboration. So we both solve problems in the communities through the traditional kind of chip activities, but we also look at ways that we can address novel and new questions through the research process. So Paul talks about having a new model of drive-by research, which is actually where you guys are able to come by the university and shop for your academic partner that you want to collaborate with. And I think this is maybe a figure some of you have seen. It's one that you can't actually probably see, but it's basically a comparison of this community-based participatory research, which is really a collaborative approach to research compared to traditional research. And what it really emphasizes is that community partners should be engaged ideally in this model in all phases of the research process, from identifying the question to figuring out How do you approach the question to even looking at the data and saying, this is what the data means, to figuring out how you're going to share the data? It's a great question. So community engaged research is kind of a generic term for academics collaborating with communities on a research project. And that's where in community engaged research there's those levels. You know, it can be a low level of collaboration where an academic might come out and collect data, to a higher level of collaboration, which is really where there has been a relationship that's been developed, and there has been kind of a co-creation of what the research question is and what the approach is. And so people would say community-based participatory research has important characteristics that really highlight kind of this equitable sharing of power, the role of the community in the process, kind of that individuals are involved all along the way. So I would say CBPR is one dimension of community engaged research if you think about it like that. Is that helpful? So with community engaged research, you know, it requires trust building, building a language for both the academic and community partners, and then recognizing this complementary expertise between the two partners, and then strategizing so that both individuals can have their needs met, you know, so that you guys benefit from kind of the expertise of the scientific community, but the scientific community also benefits because you guys know about what works and how the lay of the local land is, as well as what the important questions are for your local context. So the cool thing about community engaged research and Paul's chip to chirp process is that we can look at what the issues are on many levels in the community, and then we can think about addressing them through various different sectors. And so this is a figure that really talks about, you know, the CHIP assessment is the health issues identified by the community. So in this chip to chirp process, we're going to be looking at childhood obesity, I believe, and ways of starting to ask questions about how we improve childhood obesity. But you could actually tackle it at the clinic and hospital level, public health department, or at the community setting. And there are various ways that we deliver services to these types of individuals. And then this here is the types of research that we could use in each of these different sectors. So there are various types of research. I'm just going to highlight a few, and then David's actually going to talk more about some of the methods we use. But comparative effectiveness is this idea of looking at different ways of addressing something. So let's say we have two types of diabetes medications. And what we want to do is, in a clinic setting, we actually want to see which of these is most effective. You know, maybe there's one that you have to take only one time a month. Maybe there's one that you have to take 2 times each day. And so it's really pitting two different ways of treating the same condition against each other to see which is the best in this local context. You can do surveillance, so we could see, you know, at Warm Springs, what is the actual prevalence of diabetes? And how does this relate to maybe the physical environment, or to weight status, or characteristics like that? So surveillance is really getting a picture of the lay of the land and what the levels of the conditions are. We can do evaluation research. So in the schools, we can maybe introduce a plan to look at how do we improve access to foods for these underserved children or children who have food scarcity, and we can evaluate that plan to see if it's actually been effective. We can also do implementation research. So let's say that Lebanon, they have a pretty cool farm to school program, and what we could do is we could see, well, can we replicate that, or can we actually find ways to introduce that program here into our community in Madras? And so, as I mentioned at the start, the chip to chirp is really going to give you just enough information to really be dangerous. That's actually really what a PhD does, is it gives you enough information to start engaging in the research process and really start to think critically about kind of the world around you and how we can scientifically ask questions about the world around us. And so we have a process that starts with identifying this research question, and we'll do this through a series of ongoing activities that Paul will do, looking at developing a pilot partnership and then doing academic outreach. So how do we get academic partners who can help us refine that research question and design the study that we need to answer it? We'll do that refining through this Beyond Google. So Todd Hannon from the library is going to come out, and he is just going to help us figure out, how do you actually search for information besides just going into Google and saying, tell me about obesity, or what works, you know? And so he's going to talk about using academic search engines. We'll talk about getting grants. There'll be a panel selecting research methods, so David's going to talk about that today. We'll talk a little bit about the IRB, so history repeating. Why does the IRB exist, and what do you have to do to get IRB approval in order to actually collect data and do a study? We'll actually do a research study, so we'll talk about how you implement a project, and we'll actually, Beth Ann's an expert here, I think, by now, so we'll rely on you a lot for this. We'll talk about analyzing the data, so you might have seen p-values and confidence intervals and things like that, or how do you actually know that a study somebody has done has been effective? How do we look at that information that's presented in research papers? And then we'll strategize about ways to disseminate the findings, hopefully through both maybe academic means, but also at the community level. How do we improve and raise awareness about the important work you guys are doing?
So, Alice in Wonderland, if you don't know where you're going, any road will get you there, but when it comes to research and a lot of other things, it might take a really long time and cost a lot of money if you don't have a good plan. So at OHSU, when we get funding, we usually put in an application. If it's to the National Institutes of Health, it's usually 9 months on average before you hear back on whether you've received funding or not. So what happens is they say, okay, you're funded, and we go, oh, what did we say we were going to do? So we go run back to the proposal and figure out what we actually said we were going to do and how we're going to do it now. So we go through the proposal, we go back and look at who our target participants were. We go over the budget again to make sure everything's going to work out that way, and then do the project and how we're going to analyze the data. There's a principal investigator for every project. They're kind of like the boss person, and so it's their responsibility to go back to the drawing board and make sure everything's lined up, and assign appropriate tasks and roles to the research team that they're working with. And also to make sure they go to the IRB, the Institutional Review Board, before they start work. And I hope you guys have all watched it, but if you haven't, we have a video online of an IRB presentation. So when we go back and look at our research proposal, we're looking at the big picture and then narrowing it down. So sort of what David was just talking about, we have kind of a big picture idea, pick of the month here. So what we need to do is narrow down every step of the process and really ask who, what, when, where, and why for everything that we're going to do. So we have a really good plan set in place. Some things we think about on projects are like, what if our research subjects drop out during the middle of the study? What are we going to do? Are we going to find new subjects, or do we have a plan to try to recruit them back? Things like that. All right, so we're thinking, or you're thinking about evaluating pick of the month for the Creed pilot study. So one way that this is a lot different than normal is we know what our budget is before we've figured out what our project is, and you're kind of putting together your protocol or your project plan as you're getting ready to go to the IRB. So the project budget, we can say we want to solve childhood obesity, but we have $11,000 to do this project, so we need to figure out what we can actually do with that money. But one important thing to remember is every small project is really the piece of a bigger puzzle. So, as kind of the definition of a pilot project is you're going to answer one small project, or question that then can go lead to something bigger, and you can go get funding to do a bigger project down the road. So we need to build a protocol for this project. It's really a roadmap for your whole study. It will detail the approach you're going to take, what participants you're going to use. So it sort of goes right down through the PICO questions. So who's your population and who are these participants? How are you going to recruit them? The method, are you going to use a survey? Are you going to use focus groups or individual interviews or a mix of those? How you're going to go out and collect the data, a timeline. So what you're going to have to do is answer the 5 W's and the 1 H for each of those things. And again, having contingency plans is really important. Participant recruitment. So it looks like we'll be recruiting parents in the school system to possibly fill out a survey if that's the route that we want to go. So one thing you have to figure out is how many participants you need. And actually David will be talking about that a little bit today, and you'll probably get help from a statistician to figure out what size group you need, but I'll leave that to David for later. And it also covering whether they're representative of the whole population, because we kind of touched on that a little bit earlier. If you just have mothers who are walking their kids into school, they might behave differently in general than parents whose kids take the bus or something like that, or people in the PTA might be more involved anyway and might know about it more. And we talked a little bit about dropout. We need to build enrollment into the timeline, figuring out how you would get the survey to people and how they would be getting the survey back to you. Is it going to be paper? Is it going to be online? You won't have, if you're doing parents, you probably won't have any vulnerable populations involved. So vulnerable populations, if you remember back to the IRB presentation, are like children, prisoners, pregnant women, people who are developmentally delayed or handicapped, or economically and educationally disadvantaged. And whether you're going to offer some kind of payment to the participants to encourage them to fill out the survey, and you'll need to build that into your budget. You do have to protect individual information, and this might be something good to remember because you took a little pre-test at the beginning of this whole CREED process.
Pre-survey.
Survey, yeah, sorry. This might be something that was on that you might be taking again. So things like name, date of birth, social security number, addresses, anything that could identify Paul McGinnis as somebody that we surveyed or researched. A couple of things that don't count are like height and gender for the most part. So something to keep in mind. And when you do collect protected health information, you need a data security plan. You can't leave papers with people's identifying information on a table or on your phone or on the email. So really, if you have physical paper, you can have a locked one of those little metal boxes. We've used that in the past, or having computer protected passwords and things like that will suffice. But you do need that in your plan when you go to the IRB so that they know that you will be protecting the people that you're researching. And you have your project staff or your team. I think probably a lot of you will be working on this, and so what will happen is the principal investigator, or the PI for short, is really the person who's going to run the whole project and is ultimately responsible for everything working out and going correctly. So they need to allocate jobs to the team members. So a lot of times in a formal research situation, you'll have the principal investigator, some co-investigators, and then a project manager who is really the person on the ground getting a lot of the stuff done. For CREED, it would be, making sure we're on our timeline, getting the evaluation papers out and keeping in contact with people and going to the IRB if you change anything. Research assistants, a lot of times they'll go and actually maybe hand out the surveys or do interviews with people. A statistician to help you analyze your data at the end, but also before you get started trying to figure out how big of a population you need to sample. And if you're going to use students or volunteers is another option. Everybody on your team who actually will come in contact with any protected health data or study participants are going to have to go through training. So at OHSU it's called the Big Brain. And at OHSU we do it online. It's kind of fun. So anybody who's going to be consenting people, as I said, seeing identifiable data, the PI or co-Is would have to go through this. But if you're a study team member and you're only going to see the data after they're stripped of any identifying information, maybe you're just going to be involved after the analysis, you wouldn't necessarily have to go through all the training. This is the CREED project. So this is just a quick little screenshot of what the IRB sees when we go to them at OHSU to get permission to do this project. You see everybody who's listed as a project member and whether or not they've done all of the trainings. You can see there's an infectious agents training that not everybody's done, but that doesn't apply to this study. So, but everything that we need to do, the conflict of interest in research, which is the COIR, HIPAA training, which covers protecting personal information, and RCR.
Responsible conduct of
That's
research.
right. I was like, I know what that is. Responsible conduct of research is the third training that we have to do at OHSU. So once we get a pretty specific question down and a pretty specific research plan with answers to the who, what, when, where, and why, and we think a little bit more about our population, we get our budget all kind of in order and our timeline in order, you'll have to prepare an IRB application. And actually we need to do this pretty quickly at this point. Let's see if I missed anything. Okay, and we already know the answer to this question for this pilot project. We'll go through the Samaritan IRB. Just worth mentioning, sometimes you do need to get approval from more than one IRB. Usually go through the IRB of the institution of the principal investigator. In some occasions, I did a study once where I was working with Boston University and also with the Veterans Administration, and we needed to get approval through both of those IRBs, and it can be very difficult because they can come back and say opposite things about the same point in your plan, which can really delay things. A lot of IRBs are working to try to get like agreements together, cooperative agreements, so that, say, Samaritan had another IRB that you had to go through, they might say, you know what, we'll go with whatever Samaritan decides and give you permission that way. So that's a kind of a good development that's been happening. Okay, so once you get IRB approval, you can go out and start collecting your data, and we'll get a plan for that together, and then analyze the data. And then one of your final steps, besides looking for future funding for bigger projects, would be to disseminate the results of your project. On the academic side, it's usually in the form of manuscripts to journals or poster presentations at conferences across the country. As far as the community goes, it's traditionally getting things out in the newspaper, going to different community groups, going to policy groups to actually institute policy change. But when we're doing community-based participatory research, as we're doing here, it's usually a collaborative process, so we'll be thinking of both outcomes as we go through. And our next steps are to design your pilot study, get your IRB application together, and do the city training for the IRB, and then go ahead and do the pilot project and analyze the results. Yeah, and we're still working, Melinda and I, on some academic matchmaking to try to get some investigators who are interested in working with communities on obesity and on all of the other topic areas that your CHIP group has found to be important in your community, so that in the future, if you want to do some more projects, we'll have researchers who are ready and waiting, hopefully, to work with you.
I'd like to thank you all for having me here today. It's a real pleasure. I'm excited at the prospects for the really good work that can be done when community members such as yourselves and academic researchers like me get together to try to solve problems. So thanks for having me, and thanks for being here. Point it that way. Does anyone know what this is?
Tower of Babel.
This is the Tower of Babel. It's a famous collaboration that failed because partners didn't speak one another's language. So I think you already have the point. Paul and Melinda have made it. That's really what we're here for, is to try to avoid Tower of Babel type projects. So projects are more effective when partners understand one another's language. And I'm here today to give you a glimpse, and that's the big qualifier. It's just a glimpse of what certain researchers think about, and the certain kinds of problems and the certain kinds of approaches that a certain type of researcher might take in trying to answer questions. So we're going to talk today about certain research methods that would be commonly used in a community setting. We won't be talking about test tubes or microscopes or some of that T1 sort of research. We're going to be more on that right side of that transitional spectrum that Melinda just talked about. We'll be talking about research that uses surveys and interviews, healthcare data, community measures. And even though we're going to cover quite a lot of stuff today in a pretty short time period, I do have one disclaimer, and that is that this presentation will not make you into a professional researcher. So ostensibly, this talk is about how to select a study design or a study type. If that one of those last slides that Melinda showed you had different steps in the process. And I don't think this presentation will bring you quite there. So my real hope is that what we'll be able to do today is give you some insight into the way researchers think. And we're going to spend maybe most of the time on some basic concepts that you'll see are pretty basic. It's really not rocket science. It's science, but it's not rocket science. It's kind of, turbocharged common sense in a way. And I want you to be able to take away some of these concepts, because even though we may have fancy study designs for this and fancy study designs for that, a lot of it is really coming back to some pretty basic things. So here's what I hope we will be able to do today, is to give you a glimpse of how academic researchers think of problems and questions of interest. Give you a taste, again, the operational word here is taste, of some of the research methods or tools that we use when we work with communities. And to provide a foundation on which you can build that I hope will make it easier for you when you are working in partnership with academic researchers. So if there had been any mystery around this, or if you'd even given it any thought before, I hope that some of the mystery will go away. So the title of the talk has to do with having a tool as a hammer, and what we're going to really try to talk about today is how different problems require different tools. Different tools are good for some problems. Sometimes the two can get mixed up a bit. So let's just go through and try to illustrate and maybe belabor that point. What tool would you like here? A hammer. Which tool would you like here? Okay, bottle opener. Would a hammer work? Okay, hammer might do the trick. You get the bottle open. Okay, it might not be the best tool for the job, but it'd work. So here, maybe a bottle opener. Better way to go. What's the tool here? Okay, right, a jack. What else could you use? You're a really strong person. In a previous version of this, I had Charles Atlas there lifting up a car. I took that out. So yeah, really strong person would be a good one. Any other ideas?
Cell phone.
Cell phone, great one. Call it AAA. So it's a problem. It requires a tool. Different tools might solve that problem, and they have their relative advantages. Speed, cost, getting your sleeves dirty, different advantages. So that's the concept. We as researchers have a toolbox, and we don't want to just have a hammer. We want to figure out what's your problem, basically, and what's our problem. How are we going to solve this? What's the best tool for the job? So let's take a look at what are some of the items in a researcher's toolbox. And again, this is a certain type of researcher. A lot of what we're going to talk about today is based in the science of epidemiology. Epidemiologists argue about how to define it, so don't hold me to this. But epidemiology is the study of different exposures and different characteristics and how they relate to disease in a population. So epidemiologists study groups of people. So we're going to spend quite a bit of time with some basic terms and concepts. As I alluded to earlier, these are kind of the foundation of what the rest of this will be. So we're going to talk about something called association, and we're going to talk about exposure and outcome. This I hope you'll take away with you. Then we're going to talk about a category of study methods that are quantitative. Quantitative coming from quantity. So these are studies that rely on measuring with numbers, mathematical methods. And we're going to look at 5 different types. We're going to spend most of our time talking about these first 2, descriptive studies and cross-sectional studies, to give you an idea, again, of the way that a researcher would be looking at these problems. And then we'll kind of flash through these to give you a flavor of the different ways that we might approach problems like this. And then finally, we'll talk about qualitative methods very briefly. And these are methods that don't use numbers so much. Mostly are interested in information gathered from talking with people or observing people. So let's start by talking about association. What's association? Association is when you find things that are usually found together, like Fred and Ginger. So where you see Fred, you often see Ginger. And so that's an association. It doesn't mean, let's give some other examples, and we'll start getting some graphs here. So here's an association. We find that when more people are eating ice cream, so this graph, this would be the 0 point, and along the bottom here, this represents more ice cream consumption. This would be the 0 point, and as you move this way, it indicates more sunburn. So this graph would tell us that as more people are eating ice cream, more people are getting sunburn. So there's an association, ice cream consumption and sunburn. Another association. When more people are eating ice cream, more people are having boating accidents. So interesting. When more people are eating ice cream, it turns out there are more shark attacks.
Okay, we got to get rid of all the ice cream.
Okay, why?
It's not causing it. It's just
So
associated.
why do you want to get rid of the ice cream?
Because there seems to be more shark attacks.
Okay. And what did you say?
I said, but it's not the cause, it's just an association.
Okay, did everyone catch that? That's the point here. The point is that association by itself doesn't mean cause or a causal association. So this is a very important concept. But it's common for people to say, wow, we've got a lot of ice cream consumption, as you said. We've got to cut back on that. Maybe we'll save on shark attacks. Not likely. So what's the common factor here? Shark attacks, boating accidents, sunburn. It's summertime. So we're not going to talk about this today. We'll talk about it we will talk about it in a later talk.
The assumption is that you don't eat ice cream in the winter, and that I don't see that.
Well, so now is that the assumption? Maybe winter ice cream consumption is down here. So maybe there's still some shark attacks.
I hear the ice cream stores do really good in the winter.
There you go. Summertime is what we would call a confounder here. So we have a real association here, but it's not causal. The fancy word is causal, when one thing causes something else. So, ice cream consumption associated with overweight. Interesting. This one might be causal. That's something we want to think about. So, basic concept, basic word, association. A couple of other basic terms. One is exposure. So as I mentioned, a lot of what we're talking about today comes from the science of epidemiology. And the traditional definition of exposure in epidemiology is sort of historically related to the practice of medicine and to disease. So traditionally, exposure is a state of contact with something. It could be a germ or an infectious agent, could be radiation, could be chemicals that may have a harmful effect. The definition is the idea of exposure is used in other contexts. So it's not always it doesn't always have to be with medicine and disease. It could be any relevant characteristic, a habit, or a practice that may either increase or decrease the likelihood of a certain outcome. So it doesn't have to, an exposure doesn't always have to lead to something bad. An exposure could lead to something good. If I went back to the other graph, you could say that increased ice cream consumption increases happiness. It does for me. So that's an association, and it's a causal association in my case, but it's not a negative outcome. It's the happiness factor. So when we talk about exposure, we talk about something called an independent variable. These two things are, when we start talking a little bit more about how to analyze data and how we manipulate the numbers, this might make a little more sense in terms of what's independent. We'll get back to a graph, and I hope that'll help understand that. Another basic term is an outcome. So the traditional medical is a disease that results from an exposure. But just like with the definition of exposure, it doesn't have to be medical. So it could be any condition, could be other than a disease, could be a social economic events or circumstances. And again, the outcome could be a positive or a negative outcome, something desired or not desired. And outcomes, when we start to analyze this with numbers, are referred to as the dependent variable. So now you can start to put the two together. If you think about ice cream is independent in the sense I'm going to eat it or not, and whether or not obesity results from eating ice cream is the dependency. So the weight gain depends on the ice cream, and that's why we refer to these as dependent and independent. So let's go back to our slide here. The ice cream consumption would be the exposure. Being overweight would be the outcome. And again, this is the independent variable. As this goes up, then this one changes accordingly. So this variable, in some ways, is dependent upon that variable. The fancy word variable from your algebra class you may have skipped, or maybe not, is that a variable is something that can change. It doesn't have a set value. So ice cream consumption is something that is variable because it moves, varies by person, may vary by season, etc. So back to exposure and outcome. It's another way of looking at the same problem, and we're going to start to think of it in terms of these squares. Exposure can be, this would be the exposed group, those who are eating a high amount of ice cream. The non-exposed group, those who are eating a low amount of ice cream. So when we think about exposure, we can think about how different people may or may not have that exposure. And here we've broken it into two categories, high and low ice cream consumption. So we can start to look at differences between people according to their exposure status. Some people don't like ice cream. They don't eat it at all. Some people eat a lot of it. There may be some interesting differences between them. So that's the way we start to think about exposure. Same with outcomes. Outcome here, it's not just whether you have it, that would be overweight in this example, but whether you don't have it, whether you're not overweight. So, does everybody who eats a lot of ice cream get overweight? Well, we all know people who seem to not put anything on. They can shove this stuff down, they don't get overweight. And do all people who don't eat ice cream, are they all thin? No. So that's kind of the bread and butter, if you'll excuse the analogy, of what many researchers, especially epidemiologists, do. We're trying to figure out, are more of the people who are exposed obese or overweight than the people who aren't exposed? Are more people who don't have this outcome likely to be exposed? We try to fill these boxes up and see how they compare. So, some people who are exposed, who have high ice cream consumption, are going to have the outcome, and some aren't. And similarly, some with low ice cream consumption are going to be overweight, and some aren't. So here's the problem. Too many kids are overweight. And one other thing I'd like to say about exposure and outcome before we move on. You're going to see this slide several times, I guess. And that is that we generally think of exposure and outcome in the context of a question. So Melinda was talking about a hypothesis. And we've set this up like this is the exposure and that's the outcome. But just setting it up this way doesn't mean that this relationship exists. Okay? It's a question. We're asking ourselves, does high ice cream consumption lead to overweight? And it makes sense to set it up this way. We wouldn't intuitively say that the exposure would be overweight. Does overweight lead to higher ice cream consumption? We might find that more people who are overweight are eating more ice cream if we just looked at them. They might continue to do so, but we wouldn't think that there's a cause that way. So when we start to talk about exposure and outcome, it's kind of to set up the problem, much in the way you just said. We define an exposure for our purposes, for how we're going to set up the problem. It's kind of what we do as researchers. We set the question up, we formalize the question so that we can then answer the question. So a problem is that too many of our kids are overweight, and this isn't really phrased as a researchable question. It's just sort of a problem. But within this problem are many potential researchable questions. And what I'd like to do is take you through a couple of hypothetical researchable questions to help you see some of the tools that we use as researchers. You could start to answer a question like this using some of the lab methods that Melinda alluded to. You could do basic research in the laboratory on different hormonal mechanisms of obesity. You could look at genes. These are all things that affect obesity. The kind of stuff we're more likely to do in the community relates to working with people, working with the kinds of data we talked about earlier. So this is what we're going to talk about next. These are some of the quantitative methods we talked about. We'll start with descriptive studies, and we'll spend a little bit of time talking about cross-sectional study because I think it gives you some insight as to how we analyze these relationships between exposures and outcomes. All of these studies, incidentally, except for possibly the descriptive study, are just trying to get at the relationship between exposure and outcome, but they're doing it through different ways. And they do it because maybe there are different data available, or maybe you're trying to answer, maybe you're looking at the question with a different definition of your exposure, so you need to use a certain study design. It's kind of like a couple of ways to fix that flat tire. Are we going to use the jack? Are we going to use the cell phone? Not quite sure which. So, let's talk about descriptive studies. Here's the question. How many kids are overweight? What's the exposure? What's the outcome? There's not really an exposure and outcome in here, is it? It's really just, it's descriptive. We're asking for information to describe the way it is. So the way we set this up earlier, we had overweight as an outcome. It doesn't really make sense to have an outcome without an exposure, does it? So we don't really have an exposure in here. So as Jamie suggested earlier, or as we were talking about, we want to define our population. So we want to decide which kids. So for our study design, let's say 10-year-old children in the county. Very explicit about who we're interested in. What do we mean by overweight? We're going to measure the BMI, and we'll use a cutoff point, a standard cutoff point. And then our job is to calculate the percentage of these 10-year-olds in the county who are overweight. They're above a certain BMI. So we're not really looking at exposure in this case. We're not looking at a relationship between two things. And this is characteristic of a descriptive study. We could have asked the question, what proportion of people in the county eat a lot of ice cream? That would be a descriptive study as well. But it doesn't compare the exposure to the outcome. There's no comparison between the two. So we're going to take this. We have 600 children in the study in the community. And we go out and we weigh them. And we measure their height. We calculate their BMI. And we find that, using our definition of overweight, 180 of this 600 were overweight. And the rest, 420, were not overweight. So 180 subtracted from 600 is 420. So what percentage, that was our question we were trying to ask, trying to describe the percentage of overweight kids. And what percentage are overweight? It's the 180 divided by all of them. So it turns out to be 30%. So that's a basic idea of a descriptive study. We describe something. This could be very useful. It doesn't tell us about what might be causing the overweight, but it helps us to establish that there's a problem perhaps. Maybe the next community over is only 20%. Maybe the national average is 15%. Or maybe the national average is 40%. This gives some idea of what you're starting with in your community. Let's talk about another type of study, cross-sectional study. This study will show something about the relationship between exposures and outcomes. A different question. Do overweight kids exercise less? So what's the exposure here? No, it might be, it's kind of right here for you. The exposure here is exercise. And as I'm looking at this, I'm seeing that this could actually go in both ways, and that's kind of why this is cross-sectional. This cross-sectional, as you'll see in a minute, doesn't really necessarily talk about which comes first or what the direction is. The outcome is overweight. And when we do a cross-sectional study, we get information on the supposed exposure and the supposed outcome at the same time. So we go out and we ask people, we're going to measure people's weight, and we're going to say, what's your current exercise? Okay, we can't really tell which of these things came first, but we think, for the purposes of our hypothesis and our question, that exercising less or lower exercise may be associated with overweight. Associated, not necessarily causally. We don't know that yet. So here's our study design. We're going to measure BMIs. That's body mass index. It's a measure of one's being overweight or not. And we're going to survey people about their current exercise level. So here's our exposure again. We're going to consider an exposure, just for this example, as exercising less than 1 hour a day. We're calling this the exposure because our hypothesis is that low exercise may cause or be related to overweight. That's why we're calling that the exposure. Is that clear? We're kind of making it up for our problem. And non-exposure would be 1 hour or more a day. We're going to have two categories of outcome, overweight and not overweight. Again, we'll define it by a certain BMI level. So I'm going to move these around a little bit. We're going to move the exposure category down here. We're going to move the outcome category up here. So these are those who are exposed. These are those who are not exposed. These are those who have the outcome. These are those who don't have the outcome. And we're going to turn it into a little square. Now, I've heard this 2x2 square referred to as the epidemiologist's worldview. So if you want, in a nutshell, if you want to catch, we saw that little picture with the guy's head open. This is all we're thinking about. It's not complicated. It's a little 2x2 square. And no matter how fancy the numbers get when they're doing all this research and they're talking about p-values, we're really trying to look at the relationship between exposure and outcome. And we set up this little 2x2 square because it helps us to sort the numbers out. So let's follow through this study that we did. We went out and we talked with 100 people for this hypothetical study. And we started by weighing 100 people. And so we found that 30 of the people were overweight. And so we put those people in this category, in this column of the outcome. Again, cross-sectional study, outcome, exposure, we don't know which leads which. The other 70 were not overweight, for a total of 100. Then we asked the people who were overweight about how much exercise they got. We found this. 20 of those who were overweight said they exercised less than an hour a day, and 10 said they exercised for an hour or more a day. So not everybody who was overweight was a low exerciser. They didn't all have the exposure, but it looks like twice as many had the exposure, right? We did the same thing with people who were not overweight. And of the 70, 35 said they exercised less than an hour a day, and 35 said an hour or more. So half and half. And then, because we like to do this with the numbers and it comes in useful, we round the numbers out. So that this column, the 20 and the 10 add to 30, and this column, the 35 and 35 are 70. This is our categories for the outcomes, overweight and not. Of all the people who exercised less than an hour a day, there were a total of 55. 20 were overweight, 35 not. So, what does this tell us? It tells us a relationship between these two, between the exposure and the outcome. This kind of study does tell you something about the relationship. And we make a calculation. Here's our calculation. We ask ourselves, what proportion or what percentage of the study participants who were overweight exercised less than an hour a day? Remember, our exposure is low exercise. So we take the 20 and we divide it by the total. And we find that 20 divided by 30, 66%, or 2/3 of those who were overweight were getting low exercise. Over in this column, we did the same thing. This 35 is coming from here. It's not coming from there. It's the 35 who were getting less than an hour a day. 35 divided by the total of 70 is 50. So what does this tell us? Look at these numbers here. Someone want to tell me, what is the conclusion you can draw from this cross-sectional study?
Those that exercise less than an hour a day are more likely to be overweight.
Okay. Or put another way, those who are overweight are less likely to exercise an hour a day. Does everybody see that? There are 66%, or they're more likely to be low exercisers. Does that make sense? So 66% of those who are overweight exercise less than an hour. Only 50% of those who are not overweight exercise less than an hour. So the difference here is between 66% and 50%. More people overweight getting less exercise. Now, when we talk about statistics later on, we'll figure out whether it makes a difference, 66 and 50%. Because maybe that doesn't make a difference. What if this were 66 and this were 63? There's a difference, but maybe it doesn't make a difference. We'll get to that at another talk. So, cross-sectional studies like the one we just described have some advantages. They're pretty quick. They're pretty inexpensive. You just go out and you get the information you need all at once. You don't have to follow people through time. You don't have to go back and look at a lot of information. The disadvantage is that you don't know what came first. So we didn't really ask them about the history of their exercise. How long did it go back? We just asked, what's your current exercise? So it's cross-sectional. We're just taking one moment in time. And because of that, we can't tell about what we call a temporal relationship or a time relationship between the exposure and the outcome. It gets back to your very first question about the ice cream and the sharks. You know, we don't know if there's a causation there, but if one of them happens first, that helps us to understand that there might be some causation. All right, now we're going to go kind of quickly through 3 more study designs. I want to emphasize again that all 3 of these studies are getting at the very same sort of thing, relationship between an exposure and an outcome. But this time we're going to start seeing that time gets into it a little bit. A different question. Is playing video games associated with obesity in children? What's the exposure?
Playing video games.
Playing video games. What's the outcome? In whom? Okay, so again, we want to know which children. We want to define that. We want to know what we mean by playing video games. How often? How much do you have to play? You know, is it 10 hours a day? Is it once a month? We need to come up with a cutoff. So it gets back to Jamie's question. And again, obesity, we'll want to define that. So in this case, with a case control study, the key element is that we're going to compare the study participants according to their outcome. We call them a case and a control. Okay, case control study, we're going to say we're going to be comparing those who are obese to those who are not obese. And so we're going to identify all those kids who are obese, and we're going to identify kids who are just like them in all ways, except they're not obese. We call them the controls. We're not going to go into this today, but trying to find kids who are like them in all ways except being obese, that's the challenge in this kind of study. And then we'll get information about their prior video game exposure for the last 2 years. Okay? Here's the way it's set up. Exposure and outcome. But in a case control study, we're defining them by the outcome. So we start over here. We find those who are obese and we say, tell us about your video game use. Some of them are going to have high video game use. And that's, we go back to our question, we're saying, our hypothesis, high video game use leads to obesity. Therefore, high video game use is the exposure. And so some of them are going to have high video game use, but not all of them. Same thing with those who are not obese. Some will have high video game use, but not all of them. And like the way the numbers sorted out, we're not going to go through it again, but the way the numbers sorted out with the cross-sectional study, we'll be able to figure out, is the proportion who are obese that are on high video game use a higher proportion than those who are not? That's the essence of what's called a case control study. It's also kind of quick, relatively low cost. There are a couple of different disadvantages to this. One of the disadvantages is how you classify people. So suppose you took all of your cases, that's all of the kids who are obese, from a school that didn't have gym classes. And suppose you took all of your controls, the kids who aren't obese, from a school that does have gym classes. There's something there very important that's not similar in these 2 groups. So when you're designing these studies, don't try this at home, is what I'm trying to say. This is something you should understand about your research partners and learn as much as you can as you're working on studies with them. But it takes some training and some familiarity with what's going on. It's stuff that is very important for the community partners to understand and know about. The other issue, the potential problem here, is something called information bias. You're asking people to remember their video game use from 2 years ago. Might not be so accurate. You can have some problems along those lines. Okay, we're getting towards the end here. Cohort study is a study where you are comparing participants. Again, same relationship. You're interested in exposure and control. But here we're defining our study subjects according to their exposure status. We call them the exposed and the not exposed. So we're going to now we're going to start by identifying kids with high video game use and kids with low use. And now we're going to watch what happens through time. Now their video game use may change, so we're going to check up with them every so often, maybe every 3 months or 6 months. We're going to ask them about how much they're playing, and we'll ask them about other factors too. We might ask them about things that might affect obesity, like how much exercise they're getting, how much ice cream they're eating, things like this. So, and then we follow them for some time, and after 3 years, in this particular example, we're going to measure their weight, and we're going to have measured their weight at the beginning, and we'll find out if that has led to obesity. So here's the design. Again, we have the same exposure group and we have the same outcome group, but here, and I shouldn't have called these cases here, so that's actually an error. And now this is going to be videoed for posterity, and they're all going to say, oh, he put the cases and controls there. But this should just be the outcome. So this should be outcome and non-outcome. So for a cohort study, the lingo doesn't really involve cases and controls, so my error. But it's designed in the other direction. That's the point. So a cohort study, you're moving forward in time. Case control, you know who's obese and not. You ask them to remember what the exposure was before. Here, we take the exposure and we walk it forward and we say, oh, what happens in time? Again, everybody who's a high game user isn't going to be obese, and everyone who's a low game user is not going to be obese. It's going to split out. We're going to do the same kind of numeric analysis that we looked at before. So, advantages here. The measure of the exposure is not biased. We can define how we're going to define our exposure. We can measure it. Give them a survey. We can do it every week if you want. Theoretically, you could set up a camera in the house or some monitor on the video game, and you could get a very real measure of how much exposure they have. Different than case control, where they're trying to remember back about the exposure. Here we do know about temporality, the fancy word for the relationship in time between the exposure and the outcome, because we measure everybody at the beginning to see if they're obese. We see who develops obesity as we go forward. And we can also measure multiple outcomes. So we could measure, in addition to obesity, we could look at their grades in school, how well they're doing in school. We could look at their improved computer skills. So there could be a lot of other things you could look at. Multiple outcomes when you follow this forward, if that makes sense. Big disadvantages to a cohort study. This might not be the right way to change your tire, because it's timely. It takes a lot of time and cost. And there's a loss to follow-up, which means that some kids might move out of town. They might decide they don't want to play anymore. There's a lot of reasons that you might lose people in this study. It's a good study. It's a good study design, and it gives you some more solid information than other study designs. But it's a demanding type of study to do. Okay, this is the second to the last study design. Everybody's still with me, I hope. This is the last one that we're going to talk about that looks at the relationships between exposures and outcomes. It's called a randomized trial. Sometimes people call it a randomized controlled trial. Some people think you don't need both words. I'm one of those. The question is, does teaching parents how to motivate their children to exercise work? So there are some programs about teaching parents different ways to motivate their kids to exercise for their age-appropriate motivation. And the question that we're asking is, do those programs work? So, to do this kind of study, we would define our study population. So our study population might be, and this is a true experiment. Now that's the thing I should say. It's going to be similar to a cohort study, but it's a true experiment. We're going to define our study population. So it might be the parents of 10-year-olds, again, just to stick with that. We want to make it as precise as possible. And then we as researchers are going to assign the study subjects into what we could call an exposure and a non-exposure category. We don't really use those terms in a randomized controlled trial. It would be the treatment group or the non-treatment group or the comparison group. But the treatment is very similar to the exposure in a cohort study. The only difference is we aren't just watching what's happening naturally with free-ranging humans. Okay, we're actually doing something to them. We're saying, you guys are going to get the treatment. In this case, it's a teaching program. And we're saying, you other guys aren't going to get the treatment. And then we handle both groups the same. We handle the comparison group the same except for the treatment, so we don't want to do anything different with them. And then we measure the changes in the amount of exercise. So it looks very similar. Like I said, it's not rocket science. Here we call it treatment in this case, which is an education program. It's kind of like an exposure, except that the investigators or the researchers are making these decisions about who's going to go where, and they're doing it randomly. Okay, they're not saying, they look like somebody who would really follow the program. I'm going to put them in the treatment group. Ideally, they shouldn't know. They should be picking numbers out of a hat, so to speak, although they do it in a different way than that. So these folks get the treatment. These folks don't. We call it a placebo or comparison group. And then, just like the other study, we watch what happens. Of those who got the treatment, did it increase their kids' exercise? Did the education program increase exercise? It's going to do it in some cases, maybe, and not in others. And similarly, some of the comparison folks, for reasons we might not know or understand, are going to have kids whose exercise increases and others aren't. We'll do a similar kind of analysis. Advantages here, when you do this randomization, the flip of the coin or the pulling the number out of the hat, what it means is if you have large enough numbers, if you have enough people in the study group, that the two groups should be similar, or they should be the same in all factors that you can see and factors you can't see. So it helps to make sure that you're really looking at the effect of your treatment and that you're not looking at differences between the groups, like the one I mentioned. Well, here's a group that, for whatever reason, maybe they have better education, maybe they live closer to an exercise facility. Other reasons that might, in this case, lead to increased exercise. The disadvantages, even more than most cohort studies, although that's not always the case, usually it's more timely and costly, and they're pretty complex to do. So again, it's back to the tire changing. Which tool do you want for this job? You have to make those decisions.
You like RCT?
You like those? They have
That one.
yeah. Yeah, they're very clean. Some people would call them the gold standard in research. But it really depends on the question you're asking. Okay? It's not always the gold standard. But traditionally it is, and for certain kinds of studies it clearly is. So we've talked so far about quantitative research using data based on numeric measurements, mathematical methods. This is quantitative coming from quantity. It's these numbers. It's the 2x2 tables we talked about. I'm very briefly going to talk about another type of research that often complements quantitative research. It's called qualitative. And the root word here is quality. It doesn't mean quality necessarily as good or bad, but what's something about. And it uses data based on focus groups or interviews or just the observations of the researchers. So here's a problem. We added healthful options at the school cafeteria. We put some healthful food on the cafeteria line. And then we did a quantitative study. We measured, and we found that a minority of kids were choosing these items more than twice a week. Okay, so we found that 6 to 8-year-olds were choosing these items more than twice a week. Only 32% of 6 to 8-year-olds, 24% of 9 to 11-year-olds were choosing the healthful items more than twice a week, and only 13% of the 12 to 14-year-olds. So why weren't they choosing? It's hard to tell from the numbers, isn't it? The numbers don't really give you the whole story here. Now, there seems to be a trend. The older you get, the less likely you are to choose them, but it still doesn't tell us why. And it's hard to imagine how you could answer that with more numbers. Now, if you knew a lot about why kids choose certain things to eat, you could set up a survey maybe, and you'd have a starting point. But if you didn't know anything about why they choose things to eat, you need to find out. And so a qualitative study might be the answer. So what helps kids choose healthful alternatives in the cafeteria? A qualitative study would go in and observe the cafeteria. Researchers, often anthropologists, would take field notes, observe what's happening. They might do interviews with the students, with the cafeteria staff, with the parents, try to get some insight. We would typically record these interviews. So you do the interviews, you don't just get an idea, you actually record them. They get transcribed, and then researchers will read these transcripts, and you'll get multiple researchers who go through and analyze the transcripts, and they try to come up with some agreements on this. So You might find that there were certain themes here, like peer pressure, that wouldn't come out when you measured it. Somebody says, I kind of like eating an apple, but I get teased, maybe especially the 12-year-olds. The cafeteria staff might have some insights about, well, it depends where you put it. Put it in the front end of the line, the tail end of the line. So this is just a flavor of qualitative research. Sometimes it's the best and only method. Sometimes it isn't the right method. Sometimes it works well together with the kinds of quantitative studies we just described. So, as I promised, this is just a scratching of the surface. I hope it makes you feel more comfortable with some of the ways that researchers might think, and that it might, at the very least, be kind of a good conversation starter for you. You'll learn more as you work with researchers on your actual projects, and that's what I encourage you to do. So thank you all for having me back today. It's a pleasure to be here. We've been looking forward to coming back today, and it's fun to see you. You may recall from our previous meeting this painting. It's of a famous collaboration that failed because the partners didn't speak each other's language. It's the Tower of Babel. And as we discussed last time, to make a collaboration really effective, it helps to have some understanding of each other's way of seeing things and of each other's language. And as before, that is the purpose of our talk today, is to share a little more of how certain researchers think about significance and the meaning of the data that are gathered in the course of doing a study. Today we're going to look at a few more graphs and numbers than we did last time. It shouldn't be too bad. Mostly we're going to be getting at the conceptual stuff, but there'll be a few more, a little bit of, it won't be too technical, but there will be a little more numbers and graphs in here. To truly understand the technical side of this, you really need a course in biostatistics, which isn't that tough. It's just that it's a little more than we can do in 45 minutes today. So, as before, I have a little disclaimer here. The following presentation will not make you into a statistician. But having said that, I want everyone to feel comfortable in asking a question, because some of this might either be new or it might be something you've had before and it's just not fresh. So I'd really like it if something is not registering to say, wait a second, I'd like to stop, because we can clarify it. Let's stop and make sure that it gets clear before we move on. But it's not going to be too bad. I'm pretty confident of that. So what I hope we will do today is renew your familiarity with a few basic research concepts and terms. I hope we'll introduce a few concepts and terms that are commonly used when researchers analyze and try to understand the data from their studies. And I hope that, as before, we'll provide a foundation on which you can build, and that may make it easier for you to work in partnership with academic researchers. So it's going back to that slide of looking inside the head, trying to give you a little bit more of a glimpse of what's going inside the heads of your academic partners. So we are going to start by reviewing some basic terms and concepts, things that we talked about last time, but briefly. We talked about association. We're going to talk about a slightly different but equivalent term called correlation today. We are going to talk again about exposure and outcome and the relationship between these things. And we mentioned briefly these terms of independent and dependent variables, and we're going to go into a little more detail about how those two things are associated with exposure and outcome. And we'll very briefly go over study designs, mostly as the study designs are important for interpreting data. And so some of the new stuff we'll do today is talk about comparing groups, sampling from a population, and we'll talk about statistical significance. So here's a slide that should look familiar. We talked last time about association when two or more things are frequently found together, like Fred Astaire and Ginger Rogers. It's about a relationship, two things you grow accustomed to seeing together. Not always, but often. Another essentially equivalent term for association is correlation. There's some slight differences that aren't important for today, but essentially these things are the same. So last time you may recall we had an example where we talked about ice cream consumption and how it was associated with an increase in shark bites, and it was associated with an increase in boating accidents, an increase in sunburn. And we talked about those associations not being causal. So it seemed a little strange at first. We thought, well, ice cream consumption doesn't really cause sunburn, but the two things come together because there's this other thing, a confounder called summertime. But we did talk about a presumed causal association or correlation between ice cream consumption and overweight. So we have a graph here, and the way to read this graph is to look at this axis or this line, and as this line goes from here in this direction, it indicates an increase in the average consumption of ice cream. And the other line, as we go from here up, indicates an increase in the percentage of people being overweight. So up higher is more overweight. So today we're going to talk a little bit more about the relationship between exposure and or what we call an independent variable and the outcome, or what we call a dependent variable. You may recall that last time we talked a little bit about exposure and outcome using kind of a medical model. And that's where these terms first started to arise. So the exposure might be to a germ or to some kind of an infectious agent, and the outcome would be the disease that came from that. But we also talked about the fact that an exposure and an outcome don't necessarily have to be disease and a germ and a disease as such. It could have to do with socioeconomic status, which could be an exposure, different kinds of things or outcomes that derive from that. So in review, an exposure is a state of contact with something or any relevant characteristic, a habit or a practice that may increase or decrease the likelihood of an outcome. We showed one where it increases, but it could also be associated with a decrease in an outcome. An outcome is the disease or other condition that results from the exposure.
Or lack thereof.
Or lack thereof. So, and this may be on your quiz. So-called. Paul wants me to call it a survey. This idea of independent and dependent variable and how they relate to exposure and outcome. Here's a different example. Again, we have an exposure or an independent variable. In this case, we're looking at aerobic exercise. So someone whose average aerobic exercise is down on this side of the line would be doing less aerobic exercise than someone whose average aerobic exercise is on that side. The outcome, or the dependent variable, is cardiovascular fitness. So similarly, someone down here, lower level of cardiovascular fitness than someone up here. So what I want to try to show is the logic behind these terms of an independent and a dependent variable. And sometimes it's just a factor really, or a function of how you're setting the question up. So in this case, we're setting up a question which is, if a person gets more aerobic exercise, then does that have an effect on their cardiovascular fitness? So another way of phrasing it is that if one of the variables, in this case the independent variable, changes, does that mean that the other variable changes? So this one we change, and does it have an effect on that? This one is dependent on change in this. So let's see, here we have a level of aerobic exercise. And let's suppose that that level of aerobic exercise correlates with, or is associated with, this level of the dependent variable of cardiovascular fitness. We can plot those, the place where the two of them come together. A higher level of aerobic exercise and a higher level of cardiovascular fitness. A higher still level of aerobic exercise with a higher level of cardiovascular fitness. And I think you get the idea, you see the pattern. And eventually we get something we can make a little bit of sense out of, and we can draw a line through it. It starts to look a little similar to the previous graph where we saw a relationship where one variable, the aerobic exercise, the independent variable, increases, cardiovascular fitness increasing along with it. So when we see a pattern like this where one variable increases, the associated variable also increases, something like this, we call it a positive correlation. This may be on your quiz, Coombs survey. Yeah. So a positive correlation is one in which the increase in one of the variables, the exposure or the independent variable, it results in an increase in the other variable or the outcome variable. We could see a situation like this, where an increase in aerobic exercise could be associated with a decrease in overweight. In a situation like this, where an increase in the exposure variable results in a decrease in the outcome variable, is called a negative correlation. So still associated, there's still a correlation. It's just a matter of the way the two things relate to one another. Everybody's good with that concept? So last time you may remember we talked about a variety of study types, and this is a list of the main quantitative study types we talked about. We talked a little bit about qualitative research as well. And we talked about the fact that these different study types, descriptive study, cross-sectional, case control, cohort, randomized trial, can be used to answer different questions under different circumstances. They all have their own advantages and disadvantages. There are some sorts of questions you really wouldn't use a certain study type for. There are others that you might use several for. You might make your decisions based upon your budget or your sample size, how much time you have. So we learned that these different study types have a lot of different characteristics. One thing they have in common or several things they have in common are 4 basic stages. So you start by posing a researchable question. That's what this group's working on this morning. You design the study to get data to answer that question. These 2 things in some ways are happening hand in hand with this group. You guys are working on your study design, you're tweaking it a little bit, your researchable question is starting to focus in a little. Once you have a protocol that's based on your study design, you gather some data, and then you analyze and interpret the data. And another thing that all of these study designs more or less have in common is that they compare one group with another. So they will be looking at an intervention group, the eye of the PICO, and comparing it to the comparator group. That's why we call it the comparator group. So I'd like to take this idea of comparing groups and start to explore some of the approaches that we use and the way that we think about designing groups that can be compared, and some of the issues that a statistician and someone designing a study needs to consider when trying to compare groups. So let's start with this question of which school has the taller 7th grade boys. So I put this talk together a couple of weeks ago. It was close to President's Day, so I figured Washington Junior High and Lincoln Junior High would be good schools to look at. So we're trying to figure out whether Washington or Lincoln Junior High has the taller 7th grade boys. So we go to do our study one day, and we go to the cafeteria at Washington Junior High, and the first 7th grade boy that comes out, we measure, and we get his consent and IRB and all that. And then we measure him, and we find that he is 62 inches tall, 5 feet 2 inches. Later that week, we are over at Lincoln Junior High. We do a similar thing. First boy coming out, 7th grader, we measure 56 inches, or 4 foot 8 inches. So based on this, we can make a conclusion that the 7th grade boys at Washington Junior High are taller than the 7th grade boys at Lincoln Junior High. We see that 62 inches tall at Washington and 56 inches tall at Lincoln Junior High. And that's an important point about what we're talking about today, is this idea of sampling, getting a sample or a smaller portion of a larger group and trying to make some conclusions about that larger group based on the smaller sample. The fancy word for this is inductive statistics. So the idea of induction is making a conclusion about something more general based on something more narrow. And that's if we could, it probably wouldn't be impractical in a situation like this to line up all the 7th grade boys in both schools and just measure them. We wouldn't need statistics because we would know. We would know which is truly taller, presuming we measured accurately and reliably. But let's take this example then and expand it. Suppose I got the second boy in Washington Junior High who came out of the cafeteria that same day, and he's 54 inches tall. So this kind of is consistent with Melissa's conjecture that maybe we got a taller kid at that school. And the second kid who comes out of the cafeteria at Lincoln Junior High, 7th grade boy, 63 inches tall. So we can calculate a mean or an average. Mean is just another way of saying the average. And the average we would calculate by taking the sum or adding up all the heights of the boys who we measured and dividing that sum by the number of boys that we measured. So we would add 62 and 54, which we're doing here, and we would divide it by 2, which is the total number of boys that we measured. So 62 plus 54 is 116. We divide that by 2, and we find that the mean average at Washington Junior High, based on a sample of 2, is 58 inches. We do a similar kind of calculation for Lincoln Junior High, 59.5 inches. So now our conclusion kind of reverses. It looks like Lincoln Junior High is a little taller. Anybody happy with this? Okay, for the same reason. Okay, so let's assume there's 100 7th grade boys in each of these schools, and we sampled 35 from each. So now we've got a sample of 35. We found that the mean at Washington, same sort of a process. We take the sum of the heights of all 35 of those boys, we divide it by 35, 58.3 inches. Similar process at Lincoln, 58.1. They're starting to look a little more similar. So here, in fact, is a distribution of Washington Junior High, the 7th grade boys. And there are 100 in here. Here's the way to read this particular graph. On the bottom, we have the different possible heights, ranging from 53 inches to 65 inches. And the height of each of these bars represents the number of boys who had that height. I shouldn't use the word height. Let's just say that how far on this axis this bar goes represents the number of boys who were this height. So the boys who were 53 inches tall, there were 3. 1, 2, and then 3 is right here between 2 and 4. There were 5, right between the 4 and the 6. There were 5 boys who were 54 inches tall, etc. There's a total of 100. And I made this chart, so I guarantee you it was 100. So what we did was exactly what happened. We didn't do it by design. We went to do a sample. We took the first kid who came out, and at Washington, it happened to be somebody down here, just as Melissa suggested. It was a kid who was 62 inches tall. So you can see he was a little bit on the tall side for his total group. Over at Lincoln, relatively similar distribution, not that much different, a little bit of a difference. The distribution is the word for the way the height is kind of spread out over all the boys. So again, some of them are on the shorter side, some of them the taller side. There's a big bunch of them kind of in the middle here. And sure enough, our sample was on the shorter side here at Lincoln. So we can look at these charts in a different way. I can kind of take a look at the pattern of this graph, and it looks a little bit like this. You can kind of see if I do it fast, they kind of almost overlap. And what we're looking at here is called a normal distribution curve. So it's a way of representing the distribution, or the way that different characteristics get spread out over a population. So in this case, the characteristic is height, and height among the 7th grade boys was spread out or distributed over the 7th grade boys in a pattern that looks something like this. A small number who are on the tall side, small number on the shorter side, but the bulk in here. And this kind of a distribution is something that we find in many different natural phenomena, not just in the height of 7th grade boys. So here we have a smaller version of those two graphs for the situation we just talked about. Here's the mean for Washington Junior High. It turns out that if you took all 100 that I showed in that first graph and averaged them out, it would be 58.4 inches. And it's the exact same mean as over here. So both schools, the average for the population was exactly the same, but our sample was a little bit different. So in general, the lesson here is that we aren't ordinarily going to measure an entire group of interest. When you read this political polls where they said that, based on a sample of 1,000 plus or minus 3 points, they're making some conclusion about an entire state or the entire country. They're not going to go out and do a poll of the entire country. So we are not usually, for many different reasons, going to sample an entire population. We're going to take a subsample. And it's possible that we could randomly pick people who either represent that population. We could have picked somebody who was 58 inches if they were the first one to walk out. But we could also pick somebody who doesn't quite represent the total group. The higher the more individuals we sample, or the more individuals we get the information we want from, the closer, in general, we are going to get to the mean or the average for that group. So this idea of picking at random and the fact that we may or may not be representing the group is really an important one. So let's look at another quick example here. I think everyone, any questions at this point? I think everyone's got the idea. So just to sort of drive the point home, another question: who's taller, 5th grade girls or 7th grade girls? So we sample one 5th grade girl. Her height's 58 inches. We sample one 7th grade girl, her height's 56 inches. Our conclusion based on a small sample of 1 is that 5th grade girls are taller than 7th grade girls. Now, this doesn't make intuitive sense. It's not consistent with our experience. If you got this kind of a study and it went that much against your experience, you'd really want to look at their methods. And in this case, it's a sample size problem. They didn't have a large enough sample. So you're not convinced, I'm going to assume. I think you want to know the same things we did before. So let's take a look at the same sort of curves that we had. In this case, the 5th grade girls, you'll notice the distribution goes from 48 inches to 60 inches. The 7th grade boys, they were taller. The curve looked similar, but they went from somewhere around, I think, 53 up to 65. So similar pattern, it's a normal distribution, but different heights in general. So here, same kind of a thing, 5th grade girls, we happen to sample somebody on the upper end here. It turns out that 13% of this total group would be this tall or taller. Now, how do I know that? Well, I told you there's 100 in here. Well, I didn't tell you. There's 100 in here as well. And if we just count, we see there, of the girls who were 60 inches tall, there were 3, 1, 2, 3, plus 4 who were 59, so that's 7, plus an additional 6 who were 58 inches tall, for a total of 13. 13 out of 100 is 13%. So 13% of this group were this tall or taller. So it's not that improbable that we might sample someone who's on the tall side if we only sampled one girl from the 5th grade class. Similarly, 7th grade girls, the normal distribution looks very similar, but again, these are a taller group. They're going from 53 to 65 inches. Same thing here, we sampled on the shorter side of this group. 20% of the group could be 56 inches or shorter. That's 1 in 5. It's not that inconceivable that you could have made that kind of an error. So here's what we did. We'll put the two groups together. Our graph now goes from 48 inches to 65 inches. The 5th grade girls were here. We sampled from there. If we superimpose the 7th grade girls, we see that there is an intersection here where the two overlap. Makes sense. And we sampled on the shorter side from the 7th grade girls. If we took a larger sample of 30, and if we calculated the mean or the average, we find the 5th grade girls, in fact, are shorter. 7th grade girls are taller. 58 was the average, 54 there. So here is the same graph we just showed with the two means. The mean lay somewhere in here for the 5th grade girls, and it was just a little bit to the right of the 58 for the 7th grade girls. So, in summary, if we only sample a small proportion of a group, the sample size is less likely to represent the average of the entire group than if we have a larger sample. But we don't need to sample everyone, and we aren't going to be able to for practical reasons. And that's the job of the biostatistician. When you're working with a biostatistician designing a study, they're going to consider these factors. This gives you a rough idea of the way they're thinking. And they're going to consider a lot of other technical factors to try to consider what sample size do you need to answer your question, because you're not going to be able to talk to everybody. So, how do we know that our sample is not just a fluke? How do we know that we've got a sample that does represent the total group, and it was not just something that was a fluke of chance, like getting the tallest boy who came out of the cafeteria? Or another way of putting it is, when do we feel comfortable that our findings closely represent the real situation and are not just due to chance? And that's what I hope to talk about for the rest of the next 10 or 15 minutes. I'm going to flip a coin. So when you flip a coin, the coin can come up heads or it can come up tails. And the question that we're going to try to answer with this little study, so our study question is the coin fair? So a fair coin is a coin that one would expect to land on heads and tails with equal probability, or the same amount of times, the same frequency. So let's do our little study, and I'll start with, we're gonna get a bunch of data points. So each flip will be a data point. So it's like getting a person for your pick of the month, or for our heights. Each of these 100 students was a sample, was a data point. So first flip, it's heads. So Anyone want to make a decision about whether this is a fair coin or not?
No.
Anybody have a feeling, yes
Keep
or no?
going.
Okay, you want more data? Okay, so we'll get more data. Heads,
Keep going.
okay? So
I
you're
want to
not,
see the coin.
Janet Kay's not convinced.
She's good.
You want to what?
I want to see the coin,
Okay, how do you mean?
like up close and personal.
Okay,
Size.
okay, and that would be another study design. I mean, if you wanted to examine the coin and you wanted to make a conclusion about whether it's fair, I mean, maybe you would x-ray it and find that it's not weighted properly or something like that. But let's suppose for our purposes, we've designed a study where the only data we get are the flips that I'm doing. Okay, so we've got two, and we had heads and heads. And Janet Kay wants more data. Is anyone else at a point where they want to say it's fair or not? Okay, we'll get more data. Heads.
Oh my God.
Okay, what?
Let's make a bet. And I'll take heads.
What do you mean?
I'll
Why?
bet you lunch.
Why? Why are you saying that?
Because I would have expected a tail. I mean, I know each one is independent, each flip is independent, but over the long term, I would expect an equal number of heads as tails.
Okay.
So by the time we get 3 heads in a row, I'm starting to think I like that coin.
Okay, are
I
you
wanna
ready
buy
to,
it from you.
are you ready to come up with a conclusion about our?
No.
Okay, and does anyone else want to say, yes, I think it's fair, or yes, I think it's not fair?
I don't think it's fair because you're not flipping it exactly the same each time.
Okay, so, all right, well, let's assume, okay, it's another good point. So what does that point get at?
Your trial isn't standardized.
Okay, right, or it gets at some aspect of the way the study is designed. There might be some bias in the study, and that's a good question. If you come up with a result that just seems improbable, that's a really good and important question to look at the way the study was conducted. Let's assume that I have been doing a really accurate and balanced and fair toss of the coin in every way. Okay, are you ready to weigh in, or do you want more data?
No, because we know that the probability is one. It's just like all of those middle school and high school math problems with the flip of the coin. Even if you were to have 10 heads in the row, still on that next flip, you have a 50% chance of getting it.
Okay.
So you could do it 3 times, or you could do it 3,000
Okay,
times.
let's get more data.
Assuming the coin is fair.
Heads.
Yeah, assuming that it's
That's
fair.
4 in a row.
Okay, we're at heads.
So,
Anybody feeling like the coin is not fair?
Yeah.
If you're feeling like it's not fair, raise your hand if you're feeling like it's
Or the
not fair.
measurer
Yeah.
is not,
Okay.
the coin may be fair, but
Okay,
the
so
reporter may
another
not.
issue. Okay, so another issue with the design of the study. Let's assume the study was designed well. We've got 4 heads. I got 2 people raise their hand who are willing at this point to say, I conclude at this point the coin's not fair. Is that?
Yeah, let's count
Okay.
Kay.
So raise your hand at this point if you think the coin is fair. Raise your hand if you're willing to conclude that the coin's not fair.
If it was a
And
real
2.
coin.
Oh, it's a real
It's a real coin.
Yeah, and
But,
it's
okay,
hard to conclude
Okay.
when
based on four tosses of heads in a row, who wants to say the coin's not fair?
well, you're not allowing us
You
to
don't
tell
have
you
not.
why though, because your thumb and finger is on it in the exact same position.
Okay, let's
You're
assume.
saying-
You
If
guys
you
keep
assume
coming
that
back.
100
You keep coming back to study design
Yeah.
issues. For the study that we're doing now, the only data you have is the information about the toss, and we're going to assume that they're done fairly. These are really good points. You always want to question, but let's just assume that these are results you can trust.
assuming no other abnormalities, that everything is 100%
Right.
squarely done,
I just love the way
4 times is the difference between 4 and 0 is not as much as the difference between 80 and 0. So if you flipped it 80 times and you had heads, I would be more convinced that it was. But there's not enough of a
Okay.
difference between 4 and 0.
5 heads.
Can we see your hand?
We can look at everything after, but right now, let's assume we've got good data. Because the point I'm getting at, you guys, I love the way you're thinking about the design of the study, because there's a reason, I think, that you're questioning the design of the study.
Well,
But I'm trying to get an idea of your comfort level. At what point are you starting to suspect? I'm getting a lot of discussion, but I don't hear anybody willing to raise their hand except these two. Is any,
I'm just wondering if, when you call it heads and you flip it with the heads up, if that is making it be heads the next time too. You know
Okay.
what I'm saying?
right, so
But
are you
he's-
comfortable with
It's still
5
a
in a row
stem
heads?
design issue.
Yeah,
Who's comfortable with 5 in a row heads? Who's comfortable with 3 in a row heads?
To say that the coin is not fair.
Yeah, who's suspecting the coin?
5?
Who had suspicions about the coin when we got 3 heads in a row? Raise your hand.
I had
Well,
tossers.
you-
Okay, you voiced it, okay.
You start wondering.
Who
Yeah.
had, oh wait, but you,
But that doesn't
but
mean
you were
it's
willing to
not
get more
suspicious.
data.
And you
Yeah.
can conclude that. You could say the coin's not fair, but then once you punch the numbers, it's not going to have a statistically significant difference.
At this point.
Okay, we'll
Yeah.
get to that in a sec. So who at 4 heads in a row was willing to say, I don't think this is fair?
Well, I have to go there because you're not showing us, you're just telling
No,
us.
I know, I understand that. Believe me, I understand that point. I understand that there's some suspicion about the methods.
That's a training on the ethical conduct of research, and if you falsify data, your university can lose lots of grants from things. It actually just happened, I forget, in
Yeah.
Kansas.
No, it's a good point. And I don't take it personally either.
Yeah,
Assuming we couldn't see
Right.
you flipping the coin and all you did was just tell us.
Okay, so
We had one at
6.
4 over here.
1 at 4, we got 6. It's heads.
Ah.
Who's willing to say at this point that the coin is not fair? Okay, pretty much everybody. Now, these are great points about the methods, but let's take a look at this idea. These are the probabilities of getting consecutive heads when you toss a coin, assuming it's a fair coin. If you toss it one time, the probability of getting a head is 1 in 2. So it's pretty obvious. It can either be heads or tails on one toss. And another way of representing that is that it's a 50% probability, or 0.50. It's kind of, writing it this way is kind of the way you would write money, dollars and cents. So 50 cents or 0.50 is 50% of a dollar, 50 cents of a dollar. If you tossed it 2 times, you could either get a head and a head, you could get a head and a tail, you could get a tail and a head, or you could get a tail and a tail. So there's 4 possibilities, and only one of those had 2 heads. So the chances there are 1 in 4, or 25%. We get down here, Janet Kay was getting a little bit uncomfortable at 12.5. So that's about like the sampling we had for the tall side of the girls' distribution. 13% of the girls were taller than that. When we get down to 4 heads, there was a little more discomfort. Everyone was starting to cast aspersions upon my integrity as a researcher. They were saying, this guy's not doing something right here. And there's probably a good reason for that. And the reason was that it starts to feel like an improbable result, probabilistically. In other words, another way of phrasing it, it seemed like a result that was not likely to be just due to chance. There was something else going on here. It wasn't just chance. If it's a fair coin, then probability means more times than not you're going to get an even distribution of heads and tails. But if it was a coin like Carol was getting at, I want to see the coin, maybe it has 2 heads on it. Maybe it's not balanced properly. So if it's not a fair coin, you wouldn't expect to, you might start to see that there's something other than chance at play. There's an imbalance in the coin that's causing this. Because chance alone starts to get really, really low. We got up to 5 or 6. If at 5, we were at about 3%. So about 3 out of 100 times, you would expect to see heads 5 times in a row. And that's an important number, as we'll see in a second, between the 6 and the 3, because statisticians usually use 5% for a point where they start to think, this is probably not due to chance. There's a low likelihood, there's a chance of 3.1% likelihood that it's due to chance. We get down around here, and we're talking about 7 heads in a row is a little less than 1 in 100, a little less than 1%. So think about this next time you buy a lottery ticket with odds of 1 in 50 million. Let's get back to statistical significance. What statistical significance means is that the probability that the result you have is due to chance is low. So there's a low, the probability that this result, it's kind of confusing sometimes because we're talking about probability and chance in the same sentence. You have to pause for a second and think about it. But your results are not likely to be due to chance when you have statistical significance. Now, how low? When we say low, well, we choose. Now, when I say we, I don't mean we just decide on a whim that those of us who are going to be working on the particular project are going to pick the value. There's a convention. There's a standard that statisticians use. It's usually less than 5%. So most of the time, a statistician will feel like something is statistically significant when the likelihood that the finding was just by chance and doesn't reflect a real phenomenon in the world is less than 5%. They use something called a p-value, and you'll hear some of your academic colleagues throwing the phrase p-value around. P just stands for probability, actually. So it's just the a p-value of less than 0.05, as we see up here, is kind of a standard. Now, so let's go back to our first question, what statistical significance means. Does it mean that the findings are true? As Nancy said, well, no, not really. It doesn't really mean that they're true. It means that it's a matter of the degree of uncertainty, okay? So if it is, if we do 3 heads in a row, we're getting around 12%. That could have just happened. I mean, I've tossed a coin before and had 3 heads in a row. It happens about 12.5% of the time, okay? So it happens. Could I get 7 in a row? Yeah, I could get 7 in a row. It happens about 1 in 100 times. So if I don't have anything better to do and I just want to toss a coin a lot of times, every so often I'm going to get 7 in a row, heads. So it's not a matter of being true or not, it's a matter of the degree of uncertainty. So does it mean that the study's of better quality? It doesn't mean the study's of better quality. You can have a poorly done study that comes up with a statistically significant result. And in fact, as you all insisted on, there are times when if you have a result that just seems to defy your expectations, you might want to go back and see something about that study and look at the quality of the study. Another idea, another question is, does statistical significance mean that the findings are more important? I think importance has more to do with the real world. So this gets a little bit at what Sarah was saying earlier, and I'd like to just give one final example to drive this point home, and I think something like this might be on your quiz, your survey. So let's talk about 2 different kinds of significance. We talk about statistical significance, and oftentimes you'll hear a researcher or a statistician just use the word significance when what they mean is statistical significance. And it's because they use statistical significance all the time, and it's the kind of significance that they're mostly thinking about. But I want to show you the difference between something called statistical significance, where a finding is not likely to be just a chance finding, and significance that is real-world meaningful, sometimes called clinical significance. It doesn't have to be in a clinic, but it's real-world significance. So suppose we have a program to increase exercise in kids. We've designed this program. We have 4,000 participants. So I picked that number deliberately. It's a pretty large number. And as we learned in that first example, when you start to sample 4,000 people, you're not as likely to just get some chance findings. So we have 2,000 kids in the intervention group, and they're the kids who are going to receive this program, and we have 2,000 kids in the control or the comparison group. So before we start the intervention at baseline, each of these groups is getting 60 minutes of exercise a week. So we do the intervention. This was $530,000. It took us 18 months. Everybody was working at it. And at the end of this 18 months and all this money, the intervention group was measured, and they were getting 62 minutes of exercise a week. And the control group hadn't changed. They were still getting 60 minutes of exercise a week. And this was a statistically significant difference. So the difference between this group of 62 minutes a week and 60 was at a p-value of 0.03. Does somebody want to take a shot at putting into other words what this p-value means?
there was a low likelihood that those findings were due to the kids being of a higher income or a different school or that.
Okay, so it's not.
It was balanced.
So there's a real difference. It's not so likely that we actually just asked the wrong kids or got some wrong, you know, where these kids were actually, you know, 59 minutes or maybe they were 60. We believe that they really are getting 2 extra minutes a week. So is that significant? Well, it's statistically significant. We just showed that because it's a low likelihood that it's not a real difference. But is it meaningful to you? Do you want to put $500,000 in 18 months into getting 2 extra minutes of exercise?
Is that all you measured?
I'm sorry?
Is that all you measured?
Well, that's a good question. But to illustrate the idea of the difference between statistical and real world significance, let's just say that's all we measured. So the point here, though, is that just because something is statistically significant doesn't mean that, oh, we have to act on that now or we have to do something about that. It's a measure of the probability of your findings just being due to chance. But you still have to weigh it against the real world, and most people will probably say this is a program that we don't want to invest in. So, some implications for study design, something Susan talked about.
Just in terms of the prior slide, you are using the phrase clinically significant, which I think for community-based programs is not really a relevant phrase. I tend to
Right.
think of it as practical significance.
Perfect, a great term.
So, yeah, in that case, there was clearly a statistically significant difference, but practically, who cares if it increases their exercise by 2 minutes or not?
Yeah,
So
a good point.
just the practical significance
Yeah,
helps me.
and another alternative term is real world significance. Is it significant in the real world of what you're going to be doing? Yeah. So, as Susan alluded to, there are some implications for your design of your study that have to do with these ideas of how do you sample, who do you sample, how many do you sample, and this gets built into the question that you're going to ask. You need to work with your statistician when you're doing it. So you need to understand how large or small of a difference is important to detect. Do you need to show more than 2 minutes a week? If so, you need to try to make that clear, because your statistician can tell you how many how large of a sample you need to show that size of a difference. And then finally, how many participants do you need? So this cartoon, the man is saying, with this much grant money, the only experiment we can do is flip a coin. But I think you guys can do more than that with the money you have here. And I'm hoping that, in some ways, I hope that this presentation opened up more questions than it answered, because there are a lot of other unanswered questions about this sort of thing. But mostly I hope it'll make you feel a little more familiar with these concepts and more comfortable talking with your academic partners when you design your studies. So good luck.
So welcome everyone. My name is Susan Bankowski. Hi everybody. So today I'm talking about, I'm the chair of the IRBs for the oversight of human subject research here at OHSU, and I'm talking about the ethical oversight of human subjects research. I direct the IRBs and I also direct the research ethics program here. So I wanted to start a little bit with some background about why it is that we have the current protections that we follow currently for human subjects research. The Nuremberg Code exists as an international code from a judicial decree out of the trials that occurred in World War II with the Nazi physicians in the concentration camps. And the big elements that came out of this were that there would be, of course, since these were prisoner populations, there was no consent involved. And understanding that there has to be a requirement for voluntary consent, and that there has to be an assessment of risk and benefit. With these trials, they were looking to see how to have better ways to serve their soldier population and how to have better ways to treat prisoners, not in the good sense. And so this really didn't have a risk-benefit assessment at all. So those were the main things that came out of the Nuremberg Code. Then the World Medical Association came out with the Declaration of Helsinki, the first time being in 1964. It gets updated fairly frequently, so the last incarnation is from 2008. And this was a group of physician scientists who then have created a national code that we follow for the ethical conduct of human subject research. This brought in concepts of written informed consent, also appropriate scientific background, prior animal trials for when you conduct research. Many may be familiar with the Public Health Service's Tuskegee syphilis study. And so this was here in the United States. It was a public health trial initially that was funded by the government to look at the natural progression of syphilis in the population of black men in southern United States. And while this was a good idea from a public health perspective when it started, mid-study it was discovered that you can treat syphilis with penicillin. And so the population who didn't quite understand and wasn't fully, they didn't initially even obtain consent in a meaningful way from that population to start the natural progression study, then was kept uninformed about the fact that they could receive treatment. And so you could imagine that since this is a communicable disease, not only was that population at risk, but then also many of their contacts. And so this went on actually for 40 years, and there was an expose that occurred with respect to a New England Journal of Medicine article that examined what was going on and kind of brought this forward. At the same time, there were some studies going on at the Willowbrook Hospital for Retarded Children, as it was called at the time, in New York, and this was exposing them to hepatitis when it was expected that they would get hepatitis, and so facilitating the research in a way that wasn't fully consented with the parents. And so with all of these activities that have gone on over the past century, then in the mid-70s, as these things came to light and kind of culminated, they really felt that it was time, in light of the Tuskegee trials, to have a government look at what's going on. And so a group gathered at the Belmont Center in Maryland, and they were to consider what is it that we should be thinking of for the ethical conduct of human subject research. And out of that, in 1979 came the Belmont Report. And this is really, this is a great document. I read it every year when I teach coursework. And it sets out what are the ethical principles, the three ethical principles for the oversight of human subject research. And I'm going to touch upon those for you. From that then comes what we call the law for human subject research here in the United States, and that was to codify the principles in the Belmont Report. And then finally, as I said, the National Commission came out with the Belmont Report, and for years then, human subject research was conducted in the framework that was created at that time, and as science progressed, some events happened, which I'm going to talk about, that had us have some shutdowns of institutions in the 90s, where it was that science is such now that maybe the way we envisioned things in the 70s hasn't evolved, and we need to think about how we have oversight for human subject research. And so,
you mean that they stopped universities from conducting research?
They did. So there was a time in the 90s where some major institutions, University of Pennsylvania, Johns Hopkins, and Duke, had their research put on hold because the government was worried about the way that it was being conducted. And then at that point, there was a real shift in kind of building up even more so from the look in the 70s and 80s to now, what is it that is the best way to have oversight for modern scientific research? Great question. Okay, so the Belmont Report, I talked about this. So the Belmont Report has 3 duties that an investigator should have with respect to their research subjects. And it's important to think about when you look at the duties for research, when you think of a physician and the care that they give, this is to do no harm to the patient, that you're trying to cure them and do no harm. In research, it's a dual commitment where, although you're working in the best interests of the participant, you also have to understand that you're trying to answer a research question, and so you've got also a commitment to the science, and those have to be balanced. And so in that, you need to have a respect for autonomy, that the person can decide on their own to voluntarily participate in the research. Concepts of beneficence, where that you're really wanting to maximize benefits, and this is what I'm talking about, and minimizing harms. And then justice, making sure that you are distributing the burdens fairly, that you're selecting the appropriate populations in which to study, that you're not taking advantage of vulnerable populations. So I'm saying the IRB, this stands for Institutional Review Board. They are the groups that have the mandate for the ethical oversight of human subject research. We are tasked with reviewing research involving human subjects. Here in my IRB, performed here at OHSU by our faculty, our research staff, and our students. We protect the rights of and the welfare of the people who volunteer in our research studies. And then we protect, along with that, the confidentiality protections if it's protected health information. What is not known by many is that there's no universal law for the protection of human subjects in research. So I talked about the international codes, and while they're persuasive, here in our country we don't have a law that applies to any human subject research. And so in fact it flows from the way that we're funded. And here at OHSU and in many research institutions, we've received federal dollars to conduct research, and so we have an FWA, a promise that we've made to the federal government that will follow the laws for human subject research. It's called the Common Rule because there's many different agencies that fund research, and all of them agreed that they're going to follow this system. You'll also see, too, that the FDA has their own regulations if you're studying a regulated FDA product. And so just as I explained, this is tied to funding. And once you engage your institution or in collaborative research, if you have community partners you're working with and it's a study that has multiple individuals involved, then once those individuals are also engaged, then that's when you have this requirement of having a federal-wide assurance cover that commitment. Here at OHSU, we apply the standard to all research regardless of funding source. So then what is a human subject? It seems simple, but in fact, it doesn't end up being that simple. One thing to know is that, and we've got a definition that we work with, it's a living individual about whom an investigator, and whether that's a professional or a student, so these cover students as well, is conducting research by obtaining data through intervention or interaction with that person or identifiable private information. So an intervention is an easy thing to think about. You know, I'm coming to you and I'm taking blood or giving you a drug. Interactions, sometimes people miss that one in the sense that, you know, it could be that you're having a survey or something, interacting with them, but it could also be that you're manipulating someone's environment. You know, is it that you've, you know, changed the lighting in their setting, or that you've modified, you know, the exposure to some sort of contaminant in their work setting or something like that. So it may not just be that you're interacting with a person, but also their environment. As far as identifiable private information, you know, what is identifiable includes things, is there a linking system? It's not just you can tell who the person is by looking at the data, but also is there a coding system where someone can tell? And what's considered private oftentimes is considerable. So this is a reasonable person's expectation of privacy. And this is just a little chart for us to think about, you know, if you're having an intervention or interaction, yes, it's human subjects. If not, is there identifiable information? If not, then this isn't human subject research. And generally, it doesn't fall under the laws for protection of human subjects.
But that interaction could be that you're just talking to somebody, correct?
Definitely, it could be. It could be that you are on the corner of the street in downtown Portland and surveying people about things. Or it could be that you're studying how it is that people converse with other people, and that may be the interaction. So there are all sorts of things. It's a very broad definition, interaction, for sure. So people ask, well, then what is, if everything's a human subject, what is not? So this does not cover deceased individuals. If you're gathering health information, HIPAA would apply for the deceased, but no others. It could be that that there's some existing data, but there have to be a bunch of conditions that are met in order for existing data not to be human subject. You know, that it's data that's already exists at the time of the application, the investigator can't access identifiers, and that there's no link or code. So it could be exempt. We have categories of exemption that exist where it's, you know, it's a low risk study, but that there are certain protections that just need to be minimum. All right, so we've got yes to human subjects, but then it's got to meet a second part of the definition, which is what is research? So research is a systematic investigation, including research development, testing, and evaluation, designed to develop or contribute to generalizable knowledge. And I've included the NIH definition down here as well. What's important to note here is that you've got a plan and that you are going to follow that plan in order to create some sort of information and opinion, evidence-based decision that you think is valuable for others to know. Important to think here, so I have a public health background and I get these questions a lot. You know, some things are in the gray area. Some things you're not sure if they're going to be research or not. And this is not an easy question to answer, but you know, public health surveillance, quality assessment activities, maybe a case report. And so you really need to think about what the intent is of the activities. And sometimes things are done for a non-research purpose, but at some point during the continuum, they become research. You know, it becomes, you realize that you're onto something good and that you want to share it. But it may be that originally you were funded to do a quality assessment activity by the NIH, and they are expecting that you'll do this activity and share it with others. So it depends on what you're doing.
Let's say we were weighing and measuring children in schools.
Mm-hmm. We have studies that do that, and so it depends.
Just as surveillance for.
So yeah, the act of, if it's something that is done routinely at the schools and is being done there for the purpose of, you know, the public health activities perhaps, or that it's something that's required as a school policy, then that wouldn't necessarily be research. But let's say you're looking at, you know, instituting a policy that might be weighing 3 times a year versus once a year, or you know, does it make a difference if we separate children by gender before we weigh them, or things like that. So, and you're wanting to look at that and then help inform people as to how to have best practices in other schools, then that might be more like research. So issues in research design that the IRB considers, we want to think about the IRB review, what's required for us to conduct our review. I'm going to go over that. If we're getting permission for the potential volunteers, what informed consent would look like. Are we collaborating with anyone? There's a concept called equipoise. You know, if there's 2 arms, do we have an uncertainty between the arms, and that helps us justify the design? Is there a safety monitoring with respect to protecting privacy and safety of individuals in the study? Do we have any special groups that need extra protections? And do we have an investigator that can be compliant with all the requirements in the trial? Okay, so I have a list here. Hope it might be hard to read, but so IRB review considerations. And these are what's written into the law for us. They're the minimum that we consider, but we could consider more. So first we want to look at the design, the proposed. So when anyone creates a study, they put a plan together, and then they submit it to us, and we look at it to try to bring a group of people together and decide if we think that it meets all these standards. So we've got to consider that the risks to the subjects, and we say subjects rather than patients because they are considered volunteers, participants, subjects in a study. They're minimized. That those risks are reasonable in relationship to the benefits. So are they balanced? Can we balance out these concepts and feel like the design is justified? There's equitable selection of participants in the trial. If we're getting appropriate informed consent from them, or perhaps from their legally authorized representative if it's someone who can't consent for themselves. When appropriate, that we've got monitoring provisions for the data collected to ensure safety of the participants, and whether or not the vulnerable populations are adequately protected as well. And so, like I said, safety and confidentiality provisions are monitored throughout studies. I have been mentioning vulnerable populations, and some wonder, you know, what would that mean? We do have written into the regulations certain groups that have been identified specifically as being considered vulnerable, in that they may be vulnerable to coercion or undue influence in a trial. And so those are children, prisoners, and fetuses and pregnant women. Individuals who may have a reduced capacity to consent, that should say consent there, the terminally ill, students and employees or others that have been identified as potentially being considered vulnerable. And so any study with vulnerable populations, we want to make sure there's adequate extra provisions to consider their vulnerabilities and protect them. Payment is allowed in research, and you can compensate individuals for their participation, but it's considered compensation for time, perhaps expenses for getting to or from the research site. Sometimes it's even considered payment for their kind of hardship for having to go through the process, but it should be such that it isn't unduly influential. So you don't want a payment to be so high that it would induce a person to do something that otherwise their good judgment would make them not want to select. And so we think about the population who's being recruited, and we look at the payment and think if that's appropriate given considerations of undue influence.
And if when you're reviewing and protecting those human subjects, there's no mention of any compensation for the subjects, does the IRB wonder why?
Not necessarily. If there's no mention of payment, we assume that there is no compensation for participation. And at that point, the IRB would consider whether in the consent form it should be called out that you will not be paid for your time. And depending on the population, that may be an important thing to call out for certain. I've been talking about the informed consent process. You should know that it's a process. A lot of times there's a form involved that is kind of the guide for the process, but you can certainly be innovative in how you work with a potential participant in educating them about participating in the trial. And the goal of the process is to provide them with enough information so that they can choose whether or not this is a good choice for them. And that can come in a lot of ways. What you need to communicate, however, depends. These are the requirements for consent process, and if it's a minimal risk study, some of these can be modified or skipped altogether depending on if you are having an interaction or not with the person. So you want to certainly have a statement that this is research, that the purpose of the research, how long it's going to take, what's involved, what the processes and procedures are, what, if anything, is experimental in nature, if there are any risks or discomforts that are anticipated in your participation. These can include not just physical risks, but it could be, is there something that could be a psychological risk? Could there be a risk to identifiability? If you're studying illegal behaviors, could this affect your ability to interact with the social justice system? So risks can be broad. Benefits, if there are any. Now, it's not a requirement for research that there be benefits, quite honestly, but it is a requirement that the risk-benefit analysis be balanced and adequate. So, of course, the more risky a study, the more potential benefit there needs to be to make that balance. If there are any alternative procedures available to research, what type of confidentiality protections are in place? No exculpatory statements are allowed, so there can't be anything in the consent process that appears to limit the participant's ability to seek compensation and to sue if they feel like they've been wronged, to bring suit against the researchers. We just talked about undue influence, the other being coercion. So there can't be coercion. For me, the best way to think about coercion is like the concepts of blackmail. If you're in this study, then you'll get great care. And so, of course, you need to, or you won't get care if you're not in this study, and you can't condition anyone's care or abilities to seek care on participation in research. I always think of undue influence as an offer you can't refuse. That participation is voluntary. And then with some of the greater than minimal risk studies, you need to also consider, explaining where someone can get compensation if they feel they've been injured in research. If you have questions about that compensation, who to talk to. And as I mentioned, that your refusal doesn't put in jeopardy the benefits you receive.
Susan, who can interact with patients to get them to sign the consent form?
Researchers in the very broad sense of the word. So anybody can. and it depends on your study design, but the individuals who are obtaining consent are considered engaged in research. And so with that comes the responsibilities of being knowledgeable about human subject protections and being captured, and I say that in the way under some sort of an agreement with the government that you are under a program that has follows the principles for the appropriate conduct of human subject research. And that can be done a couple ways. But really it's that, but if you're working with a community group and they'll be obtaining consent, then you think about how it is that we get that group well educated on these concepts so they're obtaining consent in the appropriate way. I've mentioned minimal risk a couple of times. It's a really important concept because we have a few conditioning requirements on this definition. So I'll go over the definition and then let you know how we work with it. So, because like any legal term, it's clear as mud. But the probability and magnitude of harm or discomfort anticipated in the research are not greater in and of themselves than those ordinarily encountered in daily life or during the performance of routine physical or psychological exams or tests. So we call this a normal healthy person standard, being that you are comparing the risks in the research to those that happen to a normal healthy person. This is important because that means that someone who's sick can't be, it doesn't justify something more risky to them just because they're sick. That being said, it could be that because they're sick, the risk-benefit analysis is appropriate. It also, for community-based research, means that just because someone lives in a dangerous neighborhood or a neighborhood that might put them at risk, that they also should not be put to more, assume that more risk is appropriate. That being said, thinking about what's minimal risk is a difficult concept. So what you go through during a normal healthy visit at your doctor, first of all, a lot of adults, for better or for worse, don't go to normal healthy visits, but you think of your children, and the most risky thing my kids get is maybe an immunization. You know, my children, healthy children don't even get blood drawn. So I think the more dangerous part of my children's day is on the playground, driving in the car, perhaps going to soccer practice, and so, but that's the risk that we're comparing it to. What is it in your daily life that is risky? And so we use this definition to decide if something needs to be reviewed by a full board, a full committee of the IRB, or if it can have one or two trained professionals of the board conduct the review. If the consent process can be altered at all, so if something is minimal risk, then we can consider whether or not it's appropriate or needed to have all those elements involved. Do we really need to put a signature on paper at this point? Can it be an oral process? Should we use flip charts? You know, there's all sorts of ways to modify a consent process if they're minimal risk. And then the last way we do this is to think about allowable research for children. And I'm going to go over some of the children's considerations and how minimal risk comes into play for that. And that's the next slide. So the children's issues, there's a number of things to consider for research with children. First of all, every study that's conducted needs to fit into an approvable category for child research. And those are 4 categories. And for a working IRB, actually 3. Because the first one is minimal risk research. So minimal risk research we can approve. The second one is greater than minimal risk research. That has a prospect of direct benefit for the children in the study. And so this making sense, and best examples of these are, clinical treatment trials where it's a sick child seeking care and they're trying out a new treatment. The fourth category is greater than minimal risk research with no prospect of direct benefit for that for the children in the study, but likely to yield generalizable information about their disorder or their condition. So this means then that the children in this trial, if you're going to subject them to the greater than minimal risk activities, they have to be in that class of children that have a disorder or condition. And there's a lot about, what is a condition. I've seen papers written that say adolescence is a condition, so it depends on how you're looking at this. But those are the categories that we are allowed to approve in. If we think a study is a great study that should be done and we feel it meets all of our categories of ethical appropriateness, but it doesn't fit into one of these 3 categories, then we can send it to the government for special review and see if it's something they think should be done. We also think about how to select consent and or assent for children. Assent being a child's asking a child if they agree to participate. So if you have a child enrolling in a study, it's likely that we'll be asking their parents for consent to participate. And we use the distinction being that adults with capacity, you know, it's our that we say you become an adult legally by age 18. If you have capacity to provide consent, then children deserve the respect to be considered, to have their considerations worked into this, but it's not consent, instead it's assent. You know, yes, do you also agree to be in this? And I do.
Is there kind of a age where you start to ask the assent?
Yes, our policy, based on some studies that were done about children's capacity to understand questions, starts at 7.
Okay.
You know, that being said, I have children, and I always say that if you ask them, you know, do you want to be in a study where we're going to take your blood? And, you know, they're going to say no. So, you know, it's really that you have to think about kind of the whole process that you work with the parents and the children, and the educational process that you go through in a consent and assent. To that question, though, then here in Oregon, and it varies state by state for law, 15 to 18-year-olds can consent for themselves, 15 to 17 actually, can consent for themselves for medical treatment, for seeking reproductive health care, for getting treatment for addictions. And we respect those laws in a research setting as well. And so if the study involves seeking treatment for a research purpose, then we allow children to consent for themselves as well. Also,
that's only for that certain age and above.
It's for 15 to 17-year-olds, and it has to be a treatment specific, one of those specific treatment areas. Sometimes it's in the best interest of the children not to seek parental permission or guardian permission. Perhaps if you're studying child abuse and you're wanting to work with the children, perhaps it's not in their safe enough environment to ask the parents' permission for those child, and you have to meet some requirements for that. Also, if you're involving wards of the state, there are special considerations there. And then if you're conducting research in schools, there are a number of laws that apply to research in schools. All right, so one of the questions that comes up a lot is, of course, about protecting confidentiality. A tool that we have in research environments is something called a certificate of confidentiality. And this is a legal tool that comes from the Department of Justice, and it protects data if perhaps you're conducting research that involves potentially stigmatizing information. And so you could think that it's perhaps information on substance abuse, sexual attitudes or practices, illegal behaviors, and we have a number of things in the list. It even can be employability, insurability. And so if you're working in a study that has, conducting a study that has these issues addressed, it may be in your best interest to seek a certificate of confidentiality, and these protect the research data and then subsequently the participants from compelled disclosure from a government entity. So if you are in a study and they're asking you to be honest about your drug addiction, then we protect those records so they can't be compelled in court. I have a whole list here of what an investigator's role is. I'll go through them very quickly. In fact, you know, they are to comply with the provisions and the requirements of the IRB. They have to be knowledgeable about human subjects protections. They need to follow the research as it's approved. So the way the IRB system works, it's a prospective system. You need to get permission from the IRB to conduct your research. And if you want to change your research, you need permission for that. They need to only do that which they're approved to do. They need to ensure that subjects understand the research before enrolling them. They need to keep their records appropriately and report information appropriately to the IRB. Additionally, if there's anything unanticipated that occurs in your study, that needs to be tracked and reported as well. So to your question, who's on these IRB? Who are these magical people? So it has to be at least 5 individuals to constitute an IRB. Our IRBs are a lot larger here because the requirement is that you have the adequate expertise and knowledge then to review the science that is proposed. And we have, medical school, nursing, dentistry, public health programs, all sorts of engineering programs. So we have a very wide background. One, we need to have, like I said, qualified faculty. We also have to have at least one community member, when we have to have an individual who has no medical or scientific expertise. We want to have a balanced diversity on the board as well, and that's a requirement. Here they're appointed, but the IRB has the final authority to disapprove research. And this is really important because they may approve research and they may disapprove research, but if the IRB says no to a study, nobody here at OHSU can say, actually, this is a really important investigator. And we think that despite what you've said in your ethical review, that this study should go on otherwise. That's not allowed. And people, you have to recuse, have a person step out of the room if they have a conflict. So the IRB, I've told you their considerations. We look at the consent forms. We make sure all those requirements for review are met. And we look at their advertisements. If you're advertising, we look at their safety plan, if they have that, surveys, all their data collection tools. We look at all of those. And then once we feel like we've completed our rule, then we have to make our decision. And there's only a few things we can do. We can approve it as we see it. We think it's a great idea, which doesn't happen often. We can approve it with required changes. And there's two ways to do that. Either that it's, there's minimal changes, and if you just fix some of these administrative changes, it's good to go. But if they're substantive changes, and those substantive means those issues that I covered in the list of required elements, risk-benefit analysis, privacy and confidentiality, they're substantive, then the study goes back to the investigator. They have to answer those questions, and then the full board reviews their answers and then decides if they meet it. And then something can be disapproved if we really feel like there is no remedy that can make it fit within the guidelines for approval of human subject research.
How long does this process take?
We have 8 IRB meetings a month, 2 hours each. And so, any study can go through pretty quickly if it's expedited in 2 weeks, or it could take, 6 weeks or more, depending on if you have problems in your study. So I wanted to go over, that's kind of the basis and the background. I don't know if you have any questions about what I've discussed at this point.
Generally, how many studies are denied?
Not disapproved fully, not that many. It's not a common occurrence that someone has such a flawed idea that we send it home. Sometimes we'll say your application is so disorganized or messy that we think you really need to spend some more quality time with it. But otherwise, most of the initial studies are approved with some sort of change. And I'd say maybe about 30% of them get, at first pass, get deferred. It depends if there's another review group that's looked at it too. There's other groups that look at scientific quality, perhaps at the National Institutes of Health or through our Cancer Center has a scientific review board. But if it's a study and the IRB is the first group to ever see it, oftentimes there's a significant amount of work that's needed to bring it up to our standards.
What about studies that have been approved by other IRBs that also involve people from OHSU, and how do the two IRBs work together to not have to duplicate the effort?
Not always that well. So you're hitting on a point, though, where if it's conducted at multiple institutions, then everyone's IRB potentially has to look at it. We can set up agreements where one IRB can say that they agree to rely on the review of another IRB. And we are willing to do that oftentimes in minimal risk situations, less willing, of course, for greater than minimal risk. And I think that's a fairly common thing. But sometimes it has to go through 2 IRBs. Of course, everyone's trying to accommodate anything specific for local research context. So if it is that that there are some things about your population that are specific that wouldn't be known by another IRB, then that's the most important thing to be bringing up for an individual IRB's review. But sometimes it's just a matter of opinion as to how to write a consent form. Other questions before I move on to the cases? Okay, I'm going to take you through a few quick cases. Hopefully I have enough time to go through these somewhat. They basically highlight, these are real cases, some of them ended up as lawsuits, but they're real cases that have subsequently created ways that we enforce and form our protections. So the first one is a case of a young man named Jesse Gelsinger who was at research at the University of Pennsylvania. And so you asked, were research institutions really shut down? So this is one of the first cases that resulted in a shutdown. And this was a study of a young man who had OTC, and it was a viral vector study. What's important about this is that unfortunately he passed away and during the study, and it was felt that the family felt that they didn't have adequate consent. They had learned that the investigator had a significant financial interest in the trial. They had learned that there were some animal data that perhaps hadn't been incorporated into the consent process. And so this was to them the great harm. And in fact, one of the most significant outcomes of this is that we really nationally now have a system to accommodate conflicts of interest, to make sure that investigators are unbiased in their research, and if they have a financial interest, that we have a plan to manage that. Another, also a biomedical study at Johns Hopkins University, again resulting in a shutdown, and a shutdown being that the federal-wide assurance was limited or taken away by the federal government. But important to note that this means that all research at these are so significant that all research at these institutions must stop. And so she was a healthy volunteer in a study, and the investigator didn't have the appropriate permissions from the FDA to conduct the trial. She worked for him, so there were questions as to whether she was his research assistant, whether she felt like she could say no to being in the trial, and it was a bronchodilation study. They were bedside mixing drugs instead of using the pharmacy. And so all of these things resulted in a healthy volunteer dying. And this was really where we started to expand our concepts of who is vulnerable. Here this being someone who worked for the investigator and considering, did she really feel as if she could say no to being in the trial? This case is an interesting one because it's an environmental health study, and it's very difficult to study environmental elements because in order to study them, you can't alter them. They have to be present to study them. And so we have a center here for environmental and occupational research, and oftentimes, to look at how it is that pesticides affect children, you need to study children living and working around pesticides. So this was a lead paint abatement program in Baltimore, and the public health, in some ways similar to the Tuskegee trial, the public health program was setting out to decrease lead in the homes the older homes in Baltimore that had lead paint in them. But with limited resources, they were trying to figure out what's the best way to treat the biggest number of houses and affect the most children positively. And so they decided to try out, less, middle, and more abatement, knowing, in fact, that more worked. And they approached Kennedy Krieger Institute at Johns Hopkins to help them evaluate this. And this one's really important because some of the children in the study were found to have high levels of lead, which wasn't necessarily unexpected, and they were finding then, perhaps the lesser program doesn't work as well. But the participants were confused about how the research was being conducted. And they heard that Johns Hopkins was involved, and so they thought that perhaps these are medical doctors who wouldn't do anything that would be not in the best interest of the participants in the trial. And they were surprised to know that, in fact, they could still have high lead levels in the trial. And so it brought to attention how you separate out and accentuate the difference between what's being studied and what's not being studied, and expectations of performance. In fact, in the case of this, the judge in this case really said that there is a special duty of researchers to participants. It may not be the same as a provider has to a patient, but there is a duty here to think about the special needs of a research participant. The next one I think is really important because this talks about individuals' ability to control their own samples. And this was a collection of individuals in a prostate cancer registry. So they were holding samples from individuals being treated for prostate cancer. And the investigator was going from one institution to another and wanted to take his samples with them and asked permission of all the volunteers in the trial if we could do that. And most of them actually said yes. But in fact, this was an important distinction that it wasn't at that point the volunteers in the study or even the investigators' ability to decide where these samples went. In fact, they resided with the institution that funded the study. And this was a bit shocking to a lot of the people who volunteered because they thought that they could control their information and their samples as their property. But I think this is something that is little known, I think, in the general public, that when you give up your information in a healthcare setting and in a research setting, that then they rest with the institution. And I think there's a great opportunity for research in this area because we've got a couple of cases recently that kind of surround this issue, and I think there's a balance between public health and understanding ownership of samples. The next case is the Havasu Pai case in Arizona. This was a great case of participatory research at the start, where the population of Havasu Pai came to University of Arizona and Arizona State, and they said, we have a high incidence of diabetes in our population. Can you help us study this? And so they worked together to do this and then had a lot of samples. And they obtained consent from the participants, 400 members of the tribe, and had those for this study. They then shared samples beyond what was allowed in the consent form. And the samples went on to all sorts of studies, which were not things that the tribal population would have been okay with. One of them, most significantly, they did some schizophrenia studies and various other studies, but the most significant to them actually was a study of origin of the population. And so while as I understand it, this tribal population believes that they sprang from the base of the Grand Canyon. In fact, this study suggested that they came over the Bering Strait, and this was very harmful to them as a community. And so I think what's important to come out of the Havasu Pai case is that it's just not individual harms. You have to think about community harms as well. And how is it that research in this population should be considered? The last two cases are similar to the Catalona case, and I wanted to bring them up. They're both similar, Bellino and Bearder cases, where it's using samples that were collected for public health purposes. So I know I was talking about the difference between research and public health. When your children are born and they do a heel stick in the hospital and take a little bit of blood, and these are stored at the public health department. They're de-identified, so you can't tell who they are by the samples. But some research was being done in Minnesota and then also in Texas. And individuals learned of this research through various channels and really felt that this was not something that should be done. Now, legally and ethically, this is something that we can, as an IRB, approve. But at the population level, they felt that there was some sort of dignitary harm that you should have just asked. And so I highlight this because I think that, as I'd mentioned before with the Catalona case and the samples, I think we have a lot of opportunity for public education to understand the partnership with researchers and the value of research when conducted appropriately, but also understanding minimal risk and understanding the concepts of the conduct of research. So, I think this is one that's a really good example of the work to be done in education about research. So that's the end of what I have prepared. I know we're short on time perhaps, but I want to make sure I answer any questions if you have them.
My only other question is, if you are wanting to prepare an IRB application, what does it entail?
So at any institution, an IRB application is going to have a series of questions that are helping us to gather enough information to answer all of our questions that we have. So you're going to have some sort of a protocol or research plan. If you're going to have a consent process, all the documents, be it consent forms, information sheets, advertisements that are part of that consent process. If you have surveys, perhaps your survey tools, your plan for monitoring data and information and safety, all of those will need to be submitted, along with typically filling out a form that helps answer all the questions that we need then to feel like we understand how you're recruiting, how you're protecting participants, things like that.
If you've never done this before, is there someone at the IRB that can give you some pointers?
At our IRB there is. So we do definitely have, we have analysts and a number of IRB chairs that are all available to help accompany you through the process. If you're here at OHSU, we have classes that help train on that. And then again, we're always willing to train the trainer so that you can help your community partners do that as well.
You also, I know you had all these things listed for what's required in the consent form. You also have some documents that can help people, right?
Yes, definitely. We have template forms for, kind of a big consent, a minimal risk consent, and even an information sheet when you know that it's just sufficient to explain the study, but then not necessarily have a requirement for signature. Because sometimes signature is not required because the only identifier for that this person has ever been in the study would be their pen to paper. So at that point, we'd say, you might not want to have that signature because then that would identify them as being in the trial.
So you started with some historical incidents and then ended with case studies at the end, and some of them are fairly scary for population to hear about. And so what can be done to help build the public trust in research when so much is probably done the right way, but those cases become the sentinels for change.
Definitely.
That make it look worse than it maybe is overall.
Yeah, and I think what you said is important. That I think in modern times now that most research is done the right way, which is surprising, I think, to the community in some ways, where their greatest fears about their interaction with researchers and perhaps even the medical community may not necessarily be as real or as big as they fear.
Stranger.
But that being said, there in every area there can be bad actors, and so learning from those activities. In the 90s, when there were the big research shutdowns, we all kind of clenched up and got almost overly compliant, where the researchers out there would claim that we were getting too much information and too scared. And I think right now is a nice time in research oversight because we're looking at what it is as IRBs we do, and does it really achieve the goal that we're after, which is protecting participants in research. And so does all the paperwork mean anything? But to answer your question, I think the most important thing is public education. And I think there needs to be more education about research and the public's role in research. We have community members on our IRBs. There are community advisory boards that inform IRBs, researchers, and so I think these are all really important parts of modern research. Great, well, thank you very much. Thank you.